due to the time limitation, I would not explain all the models here, but simply we will have the different stakeholders, maybe community, government institute, and regional clusters, chambers, tech industry, universities, and one part. So other part we will have the developer like SMEs and SMEs here means some of the product developers and R&D centers, universities, and ICTA will mediate to under National Innovation Platform. That is a platform model, and this is the what I explained under GovTech. So we will get the problem from here, and with the help of many partner, problem will be developed. The concept here is, as you already know, human-centric design, or the what we call design thinking is the concept here. So what we need to, and to little bit of agile project management also, not isolated development, always communicating with the community, and the product will be developed. So physical location identified is the Nanasala Center. Right now we have 100 Nanasala centers, active Nanasala centers. Initially, I set up 1,000, but some of them are not working right now. And innovation labs, this is private and public labs. And also prototype center that I explained. So we will have the facility center, and then we'll get the information from the people, and then with this provide service to the universities or the vocational institute and whoever doing the research. So this is a little bit of comparison of our pro lab model, what we are working right now. And probably most innovation lab is mainly focused on the interdisciplinary research, even though we have not a name, because we need to focus. If you are taking with the technology, focus and the other verticals. If you are taking the vertical, focus different technology. So create the two things. And so in some extent, other areas, even GovTech, we don't know what is the problem. So we need a lot of input and a lot of support from there. And so I think I have used around 30 minutes, and thank you for this month, this opportunity. And I think as a closing remark, I would say I think some of the interaction with the technology faculty here and technology faculty of Uavelas and Ruhuna. I think that is the country need to have, and I think excel the technological research in the country. So where you will have, even though this is a small step towards that, you will need a lot more effort, a lot more work. And I think the technological research and innovation will be the future. Thank you very much, and we would like to have your great work in future. Thank you very much.
Thank you very much, sir, for that very informative speech. Now, I would like to call upon the Dean, Faculty of Technology, Senior Professor Sumedha Jayanati, to please present token of our appreciation to Professor Abhay Gunawardana. Thank you very much. Before we conclude the proceedings of the inauguration ceremony, I have pleasure to invite Ms. Sharina Saleh, the Symposium Coordinator, to propose the vote of thanks.
Good morning, everybody. It is a great honor and pride for me to have the opportunity for delivering the vote of thanks of the virtual inauguration ceremony of the 5th Annual Research Symposium of the Faculty of Technology, University of Colombo. First and foremost, I wish to express my sincere gratitude to our chief guest, Senior Professor Chandrika N. Vijayaratna, the Vice Chancellor, University of Colombo, for accepting our invitation and gracing this occasion. My special gratitude is extended to the keynote speaker, Professor Pradeep Abhay Gunawardena, Director, Technology Diffusion at Information and Communication Technology Agency of Sri Lanka, for your innovative thoughts supporting the theme of the symposium this year, the role of technology in interdisciplinary research. Thank you very much, sir, for your invaluable contribution to this event. We all are inspired by your great words. Also, I would like to express my gratitude to the Rector of the Sri Pari campus, Deans of the faculties, Directors of the institutes, ICC committee members, and all other invitees for their esteemed presence at the symposium. I must mention our deep sense of appreciation to Senior Professor Priyadarshini Galappathy, the chair of this year's Symposium of the University of Colombo, for her presence at the symposium today. Also, I would like to express my wholehearted gratitude for the invaluable guidance, leadership, and the tremendous support provided by Senior Professor Sumedha Jayanethhi, the Dean of the Faculty of Technology, University of Colombo, and Professor Ranjana Piyadasa, the Acting Dean of the Faculty of Technology, University of Colombo, in organizing the Annual Research Symposium 2021. A special thank goes to our plenary speakers, Dr. Ajantha Athukurala, the Deputy Director of University of Colombo School of Computing, Dr. M.K. Jayananda, Senior Lecturer, Department of Physics, University of Colombo, Professor Chandrika Nanayakara, Head, Department of Plant Sciences, University of Colombo, and Senior Professor Devak Veerakon, Department of Zoology and Environmental Sciences, University of Colombo, for sharing their valuable thoughts with us. Next, my sincere appreciation is extended to all the session chairs who will be participating in this event. I must thank the Registrar, Mr. Edward, and the Bursar, Mrs. Jayasuriya, for their support in organizing this event. Also, my special thank goes to Mr. Prabhaharan, Deputy Registrar, Mr. Dayal, Senior Assistant Bursar, and Ms. Tanuja, Assistant Registrar, for their immense support towards the success of the symposium. I wish to thank all the authors who have sent their research papers to the Symposium of Faculty of Technology for their interest in presenting research findings and contributing to this event. I consider it as a great success to accept 13 abstracts in diverse fields in technology, representing Faculty of Technology and other collaborating universities after rigorous double-blind peer review. I must not forget to extend my sincere thanks to Dr. Jayan Vevalwala, Professor Chamini Hemachandra, Dr. Kanchana Abesekara, and Dr. Hansika Adapattu for their support, valuable advices, and the guidance to make this year's symposium a success. I also like to express our sincere thanks to Mr. Manula and the Technomedia of the Faculty of Technology for their support in setting up the YouTube channel of the faculty. I would like to extend my heartfelt gratitude to the members of the organizing committee of the Research Symposium, the editor, Dr. Rohan Samrasinghe, and all staff members of the Department of ICT, and the instructors and non-academic members for their commitment and timeless effort for the smooth organization and operation of this event. A special thank goes to Mr. Harsha Kaluarachi, Mr. Rohana Vijayawardena, and the instructors of the Department of IAT and Mrs. Sonali Herath for their cooperation and support given. Finally, I would like to express my sincere gratitude again to the acting head of Department of ICT, Professor Ranjana Piyadasa, for his support and encouragement to make this event a success. Thank you.
Thank you very much. Before we conclude the proceeding, ladies and gentlemen, the inauguration ceremony of the 5th Annual Research Symposium of the Faculty of Technology, University of Colombo is now concluded. All the participants joined via the online and physically, please stay tuned. After this 10 minutes break, we will start the plenary session. Now the time is to begin the plenary session. To introduce the first speaker, Dr. Ajanta Athukorala, I would like to call Dr. Rohan Samarasinghe, Senior Lecturer, Department of Information and Communication Technology.
First, I will introduce our first plenary speaker, Dr. Ajanta Athukorala. Dr. Ajanta Athukorala, who was our founding ICT head of our department, earned his BSc in computer science with the first class honors from the University of Colombo in 1995. In 2002, he received his PhD in neural pattern recognition from the University of Queensland in Australia. Dr. Athukorala is currently a senior lecturer and the deputy director at the University of Colombo School of Computing, UCSC. He has lectured for the undergraduate, postgraduate degree programs for over 24 years. His research interest includes neural network, pattern recognition, machine learning, data analytics, computer networks, and virtualization. He is a member of IEEE and the Computer Society of Sri Lanka, CSSL. He has been a member of CSSL Council since 2014, and he is currently the Society's Vice President. Over to you, Professor, Dr. Athukorala.
Hello, everyone. Welcome to the Annual Research Symposium 2021, Faculty of Technology, University of Colombo. Today, I'm going to talk on the role of information technology in interdisciplinary research, because the symposium theme is the role of technology in interdisciplinary research. So I'm going to discuss how IT or in computing that it can relate and how we can give interdisciplinary research and some input and then some improvements, right? So I'm Ajanta Athukorala. I'm an undergraduate from University of Colombo, and now I'm working for University of Colombo School of Computing. I am the head of Department for Computational and Intelligent Systems of the UCSC and also the deputy director. I was the first acting director of the Department of ICT, Faculty of Technology, University of Colombo. So because of that, I have some relationship with the faculty. And also I'm the vice president of the Computer Society of Sri Lanka, and also the following are my research interests. Okay, now I said today we are going to talk about interdisciplinary research, but the technology, and then I'm going to address the information technology. But I would like to rephrase or change my title from the role of IT in interdisciplinary research to the role of computing in interdisciplinary research, because IT is a subfield within computing. So I would prefer to call computing because that will give the full broader spectrum. So because of that confusion, most of the people try to use that alternative ways. I will try to clarify that quickly. So before the internet era came, that is in 1991, the computing was not that very popular and not that heavily used as of today. So because of that, the computing was focusing on hardware, software, and some into the business side. So we had computer engineering, electrical engineering, more into engineering discipline, as well as from software side, computer science, and the business side, information systems. But you can see IT was not there. But after 1990s, or like post 1990s, so new subfields emerged, which is software engineering and information technology. So information technology is more refers to a business and technology perspective, whereas software engineering, computer science, more into programming or the software side, whereas computer engineering is hardware oriented. So computing is a broader discipline now. It has different flavors from science side as well as from engineering. And also some are related to more hardware. That's where like it closest towards the engineering side, but from applications and the technology side, which is more into the sciences. So there are different degree programs. With that, you can see like, just give a quick quick overview here. Computer science, software engineering, information systems, information technology, and computer engineering. Because here x-axis is more like x towards 0 is more into the theory side, hardware oriented, whereas x going towards the infinity, right, or like this direction, that is more like application oriented. Similarly, in the y direction, if it is going towards this, it is more like business oriented. If it is coming towards this, it is more like hardware again oriented. So you can see computer engineering is more like hardware and theory oriented, right? It's not much business, right? And the technology side. Whereas information technology, like more technology and some business, right? But not very much hardware and theoretical side. So that's where like software engineering, it's somewhere in the middle, right? I would say it's just like getting like not going to the extremes, right? But however, I just want to say that the computing field, like when you say computing, it is just all about like, that's where like the whole X and Y axis can cover. That's where like software engineering, computer engineering, software engineering, computer science, IT, IES, as well as there is another one called ICT, which is probably in between these two, right? Information and communication technology. So these have been categorized by the IEEE and the ACM. So these are the main computing bodies in the world, right? And recently they have come up with another 3 areas, that is cybersecurity, data science, and AI. So these are emerging fields of computing now, right? So there are, they have 5 like before 2020. So after 2020, another 3 was added, so that you can see there are 8 regions are there in the field of computing, right? So IT is just one of them. That's what like I just want to highlight with my topic, right? So I will try to address more into the computing, right? So that will be like everything can be put in there. So before coming to the topic, I would say like, if I ask from the audience, are computers intelligent? I would say like everyone say probably not. Like some people might say no, there are intelligent computers are there. By the way, computers as hardware, so they are just dumb devices. They can't do much. But the only thing is when we give instructions to the computer, of course, then it can do some function. That's where like, so computers as hardware boxes, they are dumb devices, right? So they can't do much. This is like the first sort of computer, like we call ENIAC, right? So that's like, and then it is propagated towards PCs, but of course we had mainframe and mini-frame computers, whatever computer, right? So they can't as a hardware box, so that it's a machine, there's no brain. So that's without brain, you can't function. So then we had to use our brain in order to do that. So that's where like we had to write programs. So we had to give instructions, right? That is what we call a program. And then the program instructs to do some activity, and then we can get an output. So if we look what a computer is, a computer, traditional computer, we call a von Neumann computer, because that's a name that we have given to this particular computer, right? So we can give data to the computer, and then we can give a program to do something, and we can get an output. For example, if I give 3 and 5 as the data and ask the program or the computer to add these two numbers. That's a program to tell how to add, right? And then it will provide or produce me 8. That's the output that I'm going to get, right? So the input will be doing some processing with the help of main memory unit, and then we can get the output. That's a traditional computer can do, right? So if you have something to do in that way, like if you have some data and if you can get someone to write the program to do some process or some task, so then we can get the output done, right? So just like a payroll system, right? So then you have the data, and then the payroll system has to be written, and then you can run that every month, and then you can create the pay sheets of each employee. So that's a very simple thing, right? That's not a very complicated thing. So it's just input, process, output, right? So that it's like traditional way, like you would have ingredients, and then you do some processing here. Of course, you get a output, like that is your teacup, right? So that similarly, a computer is also doing something like that. But I would like to highlight machine learning or AI, right? That's where like AI. So there, it's a bit different than here. So this is the previous discussion that one human computer. I discussed data, program, and then the output will be coming here. Data will be there. The output also with that. And then we get a program. So then someone can ask how the computer is going to work. That's what so there has to be another program here. That is the program to do this task. But what I'm saying is the output is also coming another program. So what is that? I will take an example and then tell you like what it is, right? So then you can understand that. But the data is can be something like, for example, when we give data to another program, right? A computer program, I would say AI program, so that's easy for you to understand. AI program. So then we can provide some feedback, and then we can update this the training process. So that is what we call a train. We have to do the training process with the data, right? So this output is some thing that I have in my hand, right? So which I want to get some sort of a prediction, whether it's something, this object is apple or an orange, something like that, right? Or whether it's going to be rain today evening or not, right? So that is what sort of the outcome is going to be expected. So I will take an example and try to tell you. So just say these circles are the apples, and these crosses are the oranges, right? Just, I'm taking very simple binary classification problem, right? So for easy for you to understand. So if I say this is the distribution of all the apples and the oranges, right? And then later on, I'll be giving an object, right? So I will take a different color. So this will be that particular object, right? So that's I will put a circle here, right? So that purple color thing, I'm asking what it is, right? That is my question is, what is that purple color thing? That is what the question is, right? So I can give you some data, right? So from this program has to predict, it has to predict whether it's an apple or an orange, right? So then you might be happy, right? So that's what so it's not a traditional way of applying like a payroll system, which I just told you earlier. So if the audience have mathematics people, like you would say, like if you know this particular function here, like it can be y equals mx plus c, this is a straight line here, right? So then someone can say it's above the y equals mx plus c, so then it has to be an apple, right? If it's below that, I would say orange, so then it has to be an apple, right? So that is what you can predict. If you know the function, right? So that's, if you know the function of this particular distribution, yes, you can do. But if you do not know the function, right? So then we need some function approximations, right? So that is what the machine learning or the AI is trying to do, right? So this particular output is a function, right? Which tells like this is how the data distribution, the data that is being provided to this learning problem, right? And then it can predict this particular output, what class it belongs to, right? So that is what that you can see here is a very simple one, y equals mx plus c. So that I think anyone can, by looking at that, also can see that. But then as it goes on, the complexity wise, you can see there are some oranges are here as well inside apples. It looks like apples, right? So you can see the decision boundary is very, very complicated. No one, like, I don't think mathematicians here, like in the audience, can say this is y equals whatever, right? 3x cube, whatever, 2x, right? Square minus x, like plus 10, whatever, right? So some function like binary function or whatever, right? So that you can tell. It's not easy, right? So that is what, it's a very difficult problem, right? Because we don't know, right, that particular function. If you know that, it's just no challenge at all, right? But here we have only the data, right? So that's this particular program is trying to, this particular program, right, is trying to predict this outcome or the function, right? That is what I said, it's producing in a program, right? So that is what machine learning is all about. So then machine learning or AI provides computers with the ability to learn without being explicitly programmed, because we are not programming that explicitly, right? That is, what we are saying is it has to be learned by this system, right? That's where, like, so you give data and then it will learn a model. The model can be used here with our output data. That is what we need to get the prediction. So it will predict that whether it's, if it is here, right, it says it's an apple, it's orange, right? So it can say it's an apple, right? So it's simple of that. So I'm not going to explain like the difference between AI, machine learning, deep learning, right? Because today AI is a broader one, right? So then under that, we can say machine learning, right? And then even within that now deep learning, so which is very popular, and people are using deep learning techniques to do a lot of work, right? So I will take a small example here. You can say here the robots are, okay, that's a goal, but playing with the humans. Remember, these robots are independent. They are not being programmed by remote controls of anyone here. So they are independently playing. So they have to identify their players, their colleagues, as well as their opponents, as well where the ball and which side they have to kick, right? Whether towards their side or whether the other side, so that you can see the goalie is doing a good job here to protect the ball. So these all are being trained and now they are being applied. Of course, it's not easy to play with the robots like that. But however, so it's like human brain is working, whereas that artificial intelligence is also in action. This is we call RoboCup. It's like what, 3 years back, a match, right? That after match, I think they're trying to see the quality of the machines, right? So we can see, yes, we can train up to some level, right? I'm not saying it's perfect. Like we can play with the Ronaldo and his team, right? Football match, but yes, we can do up to some level. That's where like we have to look at in the interdisciplinary, right? Like how to apply this. I will tell another research site where NVIDIA, I think NVIDIA name everyone knows, it's a big brand, right? In graphics, right? So they are working with AI now, and they are trying to look the picture perfect for video calls or video conferencing, because we know that we are also coming through Zoom to do this because of this current pandemic situation. So this is not a physical or symposium, but we are coming through Zoom, right? So video conferencing is today a major thing, right? So they are looking at how to use AI, and they are trying to develop some work. So this is like very recently, June 2021. I would recommend everyone like who are interested in looking at things happening. So go to NVIDIA or just type NVIDIA research, right? You can go to their web page. There are lots of work has been done. So I just took only one. So they are using deep learning, as I said earlier. So deep learning methods to sort of synthesize the characteristics of like the human behavior, like movements, as well as trying to reduce the bandwidth, right? Because like when we are trying to transmit the data over the internet, right? You, of course, you have faced like a lot of difficulties. Now, I think you have enough experience with respect to video conferencing with the data bandwidth. So they are trying to use less bandwidth and try to do a perfect, good quality video conference. I just run this small video. Welcome, everyone. We've got a big job ahead of us.
Our mission is to change online video conferencing, making it way better with artificial intelligence, but we've only got one shot, so let's get started. We're going to need all of you, so introduce yourselves.
I specialize in facial redirection, which uses AI to help people see eye to eye.
Facial redirection is all about eye contact and framing the face. Using a single shot, my AI technology enables meeting goers to tilt their heads and move their eyes.
I make meetings more animated, with colorful avatars ready for the big screen. Using my facial cues as a guide, watch as AI animates a digital avatar right in front of your eyes. With this technology, meeting attendees can quickly jump on a call using their polished digital alter egos. No red jumpsuit or Salvador Dali mask required.
With a fraction of the typical bandwidth, I help every meeting run smoothly. Besides help make everyone look their best, this AI technique also helps reduce the video conferencing bandwidth by up to 10x, without any jitter or lag.
Great, thanks everyone. We're going to need all your contributions to succeed at this mission. We'll meet again.
Okay, I think you had a good idea what they are trying to do. So it was very interesting, right? So that we can use less bandwidth and better quality videos, right? That we can see in the future as well, right? So this is SDK, software development kit, so that people will use this, and then we will see better quality outcomes in the future. Okay, I will take a little bit some areas, especially like agriculture area and environmental area, because that's where like the faculty of technology is also specializing, because we have degree programs, and then how we can use IT or computing now, right, into get some better outcomes. One has seen this, right? Everyone knows this, that agriculture, how we can use different techniques, different machinery, right, to get better outcomes, right? So people have changed now from classical agriculture towards precision agriculture, right? So just some technologies on the GIS, as well as computer vision, GPS, but there are so many other things can be used here as well. Because through precision agriculture, we can maximize profits, like less overlaps, reduce inputs, increase yields, and also, of course, yeah, you can reduce your stress as well, because sometimes not humans are involved, the machines are doing it, because the machine has the intelligence, right, that's given by those systems that which I told you. And then, of course, it can protect the environment as well. And of course, we can feed 7 billion or 7+ billion people as well, because that's where like we need more and more food. So I will take one area, especially to discuss, like here, drones are making a huge impact, right? And then it is embracing revolutionary strategies to produce, like increase the productivity, because drones are part and parcel of this solution to give like so many like ground-based drones are there for us to provide soil and field analysis, planting, crop spraying, which is very popular, crop monitoring, irrigation, as well as health assessment of crops, right? So that's why like I will just quickly go through this. So soil and field analysis like can be done very well with the drones, because it can start in the start of the crop cycle, right? Because 3D maps can be taken, so soil analysis can be done, right? So then we can plan the seed planting patterns, like how you should do that, because with the soil analysis you can get a better outcome or better understanding, right, before you go with that. And then of course you can plan your irrigation, so the data for irrigation is there as well. And also the nitrogen level management also can be done quite well, because certain areas might need more nitrogen than the others, so that you know that before you start that. So that's all these things the drones can provide us, like, and then the farmers or the technicians can do those work. So planting, there are drone planting systems available, because they can achieve an uptake rate of 75%, as well as they decrease the planting cost by 85%. That's what people have shown, that figures, so that that some systems are there to shoot pods, right, and then the seeding with the seeds, so that we can plant that in a huge, massive yards, massive farm yards, so that that is possible. Yeah, crop spraying, I think you might have seen, this is like a lot of people are using, right? that, of course, you can correct amount of liquid or the fertilizer or the pesticides can be put in, right? So that we can monitor and we can spray at the real time, and even we can see the coverage, right? So that this efficiency is very, very, very increased, and we can reduce the chemical usage to the groundwater, right? That is very important. And also people have shown that this aerial spraying can be completed 5 times faster than we do normally, right? So that's where, like, so these are, we can do well, as well as it can give us much more efficient and good output, right? And then it can reduce the wastage as well. For irrigation, we can take hyperspectral or multispectral thermal sensors are available, so that you can see very nicely the parts of the field which are dry and then which need improvements, and then you can apply those techniques. That's where, like, agriculture knows what to do that, but of course that insight can be given by these drones, and then we can use some AI techniques to analyze and provide you insights. Like that's where, like, the relative density and the heat health of the crop can be given, and then the heat signatures can be provided. So that's where, like, in the irrigation side. And the crop monitoring also, in the vast fields, it's a very low efficiency in the crop monitoring. But there are drones, there are different types of drones, like just go and search on Google, like you can see there are different types of, like, sometimes we have seen these the drones which is in this particular picture, but there are different types of, like, drones that they have more capability and handling capacity, so that they can be used for crop monitoring. So today, time series animations can be shown, and then precise development of a crop, and then the production inefficiencies can be monitored. So everything can be done, right? So before you harvest. And also the health assessment is also very important. So we can spot the bacterial or fungi, because with the cameras today, we have very high precision cameras, very good quality pictures can be taken just in a normal mode, or like even in hyperspectral, like in a more other different modes, like EM frequency levels. So then we can analyze them quite well, right? And so that we can apply some of the, again, machine learning AI techniques to identify those areas where some assessment or like some treatment is required very quickly, and then we can apply through the other drone system, so that we can have nice drone systems, like integrated, like some are doing crop monitoring, some are doing health assessment, some are doing spraying, so that you can see, like, they can be integrated, and then whole systems can be built. So there are a lot of opportunities for us to do in these areas. So that's like, I just took some examples from the agriculture area, and then in the environment field as well. So there are this tracking water quality. Like these are kind of robots, like these are not drones now, right? The kind of robots, you can say it's like an E-like robot, like it's going through, because untreated sewage, agricultural runoffs, like heavy metals, that will contaminate the lakes, rivers. So then it's like invisible, it's kind of toxic soup, it's everywhere. So these kind of bots, like E-like bots, can swim through, and then they can collect the water samples and test them, right? And then they can provide the information back to the control station. From the control station also can analyze further. And then sometimes if you send something, right, it can swim through and follow where the toxic current is coming to it, and then we can reach through the source. So that is very important. Like most of the time, we know something has been contaminated, but the finding the source is very difficult. But these technologies can provide for the environmental, the people who are involved with them, to use these systems and get much more insight and then take necessary action. Yeah, this is also another research done by University of Bristol, where it's pollution eating robots, like it's clean up our planet, the waterways. Just like this is a swimming, again, machine like robot, it's been called ROWBOT, right? You can search for that. And then it digests pollution and turns into electric field, right? So it's like it can take the microbial fuel and then sort of like it eats and it produces, right? So similarly, you can think of like when oil spills are happening. So can we put a robot like that or bot like this and to go and eat that energy and then produce to something, right? Because then we know like it just not only produce something, but also it will just remove the oil spill that is there as well, right? So because this particular bot like that, this University of Bristol people have done, so that energy is being powered, the propulsion and the pedals through the boat to move through that microbacterial fuel cells that it will eat through that, right? So that's like quite interesting and quite good work, right? So that's why like those things can be done. And also the robots are there, like you can say, has said as environments like chemical, biological, radiological, or nuclear, or explosive, whatever you name it, right? Very high risky, so that we can use robots. And then with just, again, hardware, I said it's a dumb device, right? So we need software to run them, right? Well, that's where like machine learning AI comes in. That's where like AI is a big thing. So like even in the recent past, IEEE ACM has recognized that is as a part of the computing discipline, right? So it's very important. And I'm not going to discuss, like there's so many areas where robots are being used in hotel industry, like today, like medical, because of the COVID, like people can't reach, you can see the PCR test can be done by the robots, right, rather than humans, right, so that we can reduce the contamination. So there are so many things. And of course, we do have Sri Lankan humanoid robot as well called DSN, right? That is from Erimac. So that's also another important thing. So like you should have a look out to that as well, right? So what we can do with them and what opportunities we can get it. And also like just another example, because of the environment, we know like human elephant conflicts are there, right? How to stop that. There are so many solutions. Just one thing I have put here, just try to put like sensors and then try to detect before they reach to the human habitat areas, right? So then you can take action. There are so many ways. There are different colors, and even Sri Lanka, like in University of Colombo, also we have done a lot of work. But however, there are a lot of opportunities, and there are still problems are there, so we need to solve them. So computing, I would say, is very important form of, it's a backbone of today's digital economy, as well as from sustainable development goals as well. Like it's provides like very important activity, right? So I just a quick video, like just let's let.
Presenting the ITU Global Study on the impact of ICTs on the achievement of the UN Sustainable Development Goals. The book, ICT for SDGs, ICT-Centric Economic Growth, Innovation and Job Creation, addresses the opportunities information and communication technologies offer and provides insights and direction on how to encourage and facilitate collaborative digital entrepreneurship. It offers practical examples to leapfrog the innovation gap to ensure sustainability and create new job opportunities. The study offers a roadmap with practical strategies for utilizing advances in ICTs to foster an environment that nourishes ICT-centric innovation, economic growth and social change, consistent with the Sustainable Development Goals. Its holistic and integrated view makes the study a comprehensive resource for all stakeholders within the ICT ecosystem, including policymakers, regulators, operators, academics, funding agencies, regional and international organizations, financial institutions, and citizens to encourage collective action towards sustainable development. ICT for SDGs. ICT-centric economic growth, innovation, and job creation.
Okay, I think that's what I will not go beyond because my time is over as well. So I would like to say that, yeah, computing can do a lot into interdisciplinary research, and it's been done, right? And then people who are in the other fields should try to use this, right, and try to go to the next levels that in your fields as well, because computing can help. So computing is a supporting field, supporting discipline for most of your other areas. It's where like, so it's just not only computing people can do this, like others has to get in. So that's where like I was just trying to highlight those things. So since my time is also going up, so I will stop here. And thank you for inviting me, for the organizing committee, for the this symposium, and wish you all the best. And if you have any questions or anything, that I will try to address after the session is over. Thank you.
Thank you very much, sir, for that insightful speech. Now it's the time to present token of our appreciation to Dr. Ajanta Tukorala. The next speaker is our very own Dr. Kitsiri Jayananda. I'm extremely honored and privileged to introduce Dr. Jayananda today. Dr. Jayananda is a senior lecturer in the Department of Physics, University of Colombo. He earned his BSc Special Degree in Physics in 1984 from University of Colombo, and then he pursued his PhD in the area of experimental high-energy physics from University of Petersburg, USA in 1991. Dr. Jayananda has worked in experiments in a few major laboratories such as Brookhaven National Lab in USA, Rutherford Apperton Lab in UK, and CERN Geneva. Currently, Dr. Jayananda involved in the CMS experiment at CERN. Ladies and gentlemen, please join with me for a warm welcome to Dr. Jayananda to deliver his speech. Now it is over to you, sir.
Good morning. First, I would like to thank the organizing committee for inviting me for this talk. And as someone who has been involved in the initial stages of this faculty, I'm extremely happy to see the level achieved by the faculty in the past few years. I congratulate Professor Jayaneti and the members of the faculty for your success within this short period. The theme of this symposium is the role of technology in interdisciplinary research. So I decided to talk about my current field of research, which is experimental particle physics, and I will discuss our relationship with technology, and in particular, I will mention a few contributions made by a Sri Lankan team towards a high energy physics experiment. Particle physics is not exactly a multidisciplinary research. Also, our work is not application oriented. We are doing highly fundamental science. But in our field, we use many different technologies, and also we keep pushing the frontiers of technology to suit our extreme requirements. As an example, I have mentioned a few here. The World Wide Web is used by everybody now, but it was initially developed for particle physics researchers to communicate among large collaborations. Then the superconducting magnet technology was pushed by particle physicists at a time when they found that the available magnetic fields from conventional electromagnets was not sufficient for building new accelerators which required extremely high magnetic fields. Now these superconducting magnets are being used in many applications such as MRI machines. Accelerators are normally used for particle physics research, but now they have found their way into hospitals where they are being used for treating cancer patients. And also there are certain medical diagnostic tools such as positron emission tomography or PET, which uses particle physics detectors. In this technology, the patient is injected with radioactive material and it decays inside and radiation comes out. Particle detectors are used to detect this radiation and then in a computer to construct an image of the inside of the patient. Now, computing grids are also being pushed by particle physics laboratories in order to analyze large amounts of data generated in particle physics experiments. I'm sure these computing grids will find lots of applications in other fields of science. So what are we doing in particle physics? I will start with the atom. We know in the atom there are electrons, protons, and neutrons. And it has been found that inside protons and neutrons, there are another type of particles called quarks. And there are a number of types of quarks, although in the proton and neutron only two types exist. Each proton has 3 quarks, 2 up quarks and 1 down quark. And each neutron has 2 down quarks and 1 up quark. But by looking at various other particles that we can detect in cosmic rays and other particle physics experiments, theorists have developed a model for describing the construction of matter. This is now known as the standard model of particle physics. According to this model, there are 6 types of quarks and 3 types of particles called leptons. The electron familiar to us is here, but there are other particles. In addition to these 12 particles, there are antiparticles. These particles interact through certain interactions. So there are 4 fundamental interactions identified so far. The gravitational force is very familiar to us. That is the one that bounds planets into stars and also solar systems into large galaxies. Electromagnetism exists in atoms. In atoms, electrons are bound to the nucleus through the electromagnetism, and also all of the chemical processes happen due to the electromagnetic force. The strong force cannot be seen easily in everyday life. That exists inside the nucleus. It binds protons and neutrons together, and also inside protons and neutrons, they bind quarks together. The weak force was introduced in 1930s when they found a type of radioactive decay called beta decay. In order to explain the process of beta decay, it was necessary to introduce a force called weak force. Sometimes strong force is called strong nuclear force and weak force is called weak nuclear force. And there are 4 other particles listed here. These are called force carriers. The photon, which is familiar to us again. Light behaves like particles and waves. When it is behaving like a particle, we call it a photon. The photon is considered in the standard model as the force that carries the electromagnetic force. Similarly, the gluon is the particle that carries the strong force. It kind of glues together the quarks inside protons and neutrons. The weak force is carried by 2 types of particles called Z boson and W boson. And this last particle, which was discovered in 2012, although it had been predicted many years ago. So this completes the standard model. Higgs boson is needed to explain how certain particles acquire mass. So it seems that this standard model is extremely successful in describing many of the things that we observe in nature. However, now we are seeing certain problems, certain insufficiencies of fundamental standard model. So we are now pushing towards more experiments in order to understand certain unanswered problems that exist in this field now. There are things like why do we see more matter than antimatter? What is dark matter? Astronomers tell us that there should be something called dark matter, but we don't know exactly what it is. And there are many other things that are unanswered in particle physics at present. So how do we do these particle physics experiments? Mainly, the tool we use is an accelerator. An accelerator produces two high energy beams and we collide them together, beams of particles, for example, protons. When these protons collide, they create many new particles. By studying these, we can come into conclusions about the behavior of various particles. This picture shows an accelerator. In fact, you don't really see the accelerator. What is shown here is an aerial photograph of the Geneva area in Switzerland. There is a red line here separating the France on the left-hand side and Switzerland in the right-hand side. This structure here is the Geneva airport, and this is Geneva Lake. And this circle is called the Large Hadron Collider, maintained by the particle physics lab called CERN, European Center for Nuclear Research. The accelerator is about 27 kilometers in circumference, and it exists underground. You cannot really see it from surface. And this is the structure. About 100 meters underground, we have this large tunnel, and there are access routes through vertical tunnels. And this picture shows inside the accelerator in this part of the tunnel, but this area where experiments are conducted, the space is larger. There are actually 4 major experiments in this accelerator called CMS, LHCb, Atlas, and Alice. We are involved in the CMS experiment. And this is the view of our detector in the CMS experiment. This is a very large structure. The height is close to that of a 5-story building. It's an extremely complex detector with many different sub-detectors. And the protons go through the axis of this detector. Somewhere here, they collide, produce new particles, and new particles go through these detectors and get detected. And this is a cutaway view of the detector showing different components. And this picture shows a reconstructed image, reconstructed by a computer. The image was produced by taking information coming from various detectors in this system. The lines here show the paths of particles, and the red areas show the amount of energy deposited by these particles in the detector. And this shows a cutaway view again. Certain particles produce tracks in the silicon detectors very close to the center where this collision happens. And there are detectors called electromagnetic calorimeter, which detect certain particles. Hadron calorimeter detect another set of particles. They measure all the energy. And there are particles called muons, which don't stop in this region. They travel through a lot of matter very easily, and we have muon detectors mounted in this region to detect those. And from Sri Lanka, 2 universities are involved, Colombo and Ruhuna, 2 senior scientists and 2 graduate students from each university, and we get funding from the Ministry of Research and Innovation. This shows the picture of the control room of CMS. This is on ground, while the detector itself is 100 meters underground. When the experiment is running, we have to take data 24 hours continuously, and people must be here continuously in order to control the detector. But we cannot afford large number of people in this control room, so only a few people has to control the entire detector, which is an extremely complex one, and they don't have often the expert knowledge on all components of this detector. So we need a way of controlling this detector by non-experts. For this, we use a kind of software called SCADA for supervisory control and data acquisition software. This is a type of software used by various industries for controlling factories, and this is used by for applications such as controlling oil wells and also monitoring the harbor operations. And this system can handle large networks. Different components in the control system can exist in different parts of a widely varying network. And we are involved in the CMS detector in the sub-detector called gas electron multiplier, or GEM. This detector has a principle somewhat similar to the Geiger counter, where there is a gas filled inside the detector and high voltages are applied. For this one, there are a number of layers, and we have to apply different high voltage values, and the region here between these plates is kept with a special gas mixture. And this is a completed GEM detector module. There are many modules mounted in the CMS experiment, and the construction of this detector is a very delicate process which has to be done inside a clean room. And this picture shows a number of detectors here, a number of panels mounted in the main detector. We have a total of 144 panels like these mounted on 2 sides of the detector. So this is here there are 72, and the opposite side of the detector there are 72 more. And this is the area where they will exist when the experiment is run. For handling this detector properly, we have to provide power. And there are a number of chips here which collect data from the detector and send through this optical link to the data acquisition system. And we have to provide low voltage for the operation of these chips and high voltage for the detector to function properly. So we have to control all these. And in addition, we have to maintain the proper gas mixture, argon and carbon dioxide. We have to monitor the ratio and also the pressure. So these are the things that need to be controlled when we run the experiment. And one of our contributions to this experiment is maintaining this development and maintaining this control system. So this is a panel developed by our graduate students. This shows all the panels mounted in the system, and by going into these different panels. We can click on them and set various voltages and pressure values for them, and also monitor things like temperature and gas pressure. But operation of this is extremely complex. Only people who have intimate knowledge about the detector can control this detector using this panel. So there is a need for another way of controlling this for the people in the control room. So the solution for that is called a finite state machine. Finite state machine is a component developed by CERN. It doesn't come in the main software, the SCADA software that we buy from an external provider. Let me briefly explain how a finite state machine works. We normally teach this concept in digital electronics courses, and we often take a traffic light system as an example. So this traffic light system exists in a number of different states, and it jumps from one state to another in a particular sequence. So this concept can be developed for controlling even very complex structures. In our case, we have the main detector control system for the CMS experiment, which has number of components like vertex detectors, tracker detectors, muon detectors, and so on. Each of them have high voltage, low voltage, cooling, and gas, and so on for monitoring and also controlling. Let's take one example. If we take one GEM module that we construct, it can exist in a number of different states. It can be on, it can be off, or it can be standby. And when we turn voltage on or off, it goes through a short process. So there you can have intermediate states called ramping up and ramping down. In addition, it can go to certain error states. So we have to handle all these, and in order to change states, we have implemented a set of commands. So an operator can give commands like these: turn on, turn off, or go standby, in order to change the state of the system. Now we have to combine all these into a single control system. So the entire GEM system has two parts, NCAP plus and NCAP minus. Each NCAP has 36 super modules, which have two layers to make 72 detectors. And each module has low voltage and high voltage. We can control this entire system using a panel like this, but the operator can give a command, something like on or off, and that command gets propagated all through this detector system. These are actually called control units. They are in a kind of virtual system, and actual work is done by power supplies attached to these. So it is something like our school system, where maybe the Minister of Education says, start all schools, to his Secretary of Education. Then the Secretary sends commands to provincial directors. The provincial directors send commands to zonal directors, and so on. And finally, the command comes to the school principals. They work with teachers and open the schools and report back. Now school is open, then it goes to zonal director, provincial directors, and then to the Secretary of Education. Finally, he would announce that all schools are open. So it is something like this. When these detectors are turned on, the operator gives only one command. Everything is propagated and the right things happen. The operator doesn't have to know all the details of operating these and setting these voltages and pressure values. If something goes wrong, there are error states. The error states happen due to low voltage problems, high voltage problems, and so on. If it is only one or two detectors that go into error state, that doesn't propagate all the way. So still the full gem detector may be up and running. But if there are majority of detectors being in error states, then we have to stop the detector. So this error, we take certain majority decisions, and finally that information is propagated all the way up, and finally the gem could be down. So there is a system implemented like this, so that even an operator who doesn't know anything about the gem detector can run the system easily. So I hope I have given you a basic understanding of how a particle physics experiment works. In conclusion, I would like to mention that although particle physics is very fundamental research, we do make contributions to the society through various developments in new technology. For our work, we require cutting edge technologies, and we often find pushing the boundaries to develop new technologies. Later, these new technologies find ways to normal life of people. Thank you for listening.
Thank you very much, sir, for that informative speech. Now, the time is to present token of our appreciation to Dr. Jayananda. Thank you. To introduce the next speaker, Professor Chandrika Nanayakara, I would like to call upon Dr. Jainive Valvala, the Head, Department of Agricultural Technology.
Thank you, Dr. Atapattu. It's an honor for me to introduce our next plenary speaker, Professor Chandrika Nanayakara, to Annual Research Symposium, Faculty of Technology 2021. Professor Chandrika Nanayakara received her special degree in botany from University of Colombo in 1993 and then joined the Department of Botany as an assistant lecturer. In 1994, she was absorbed into the department cadre of the department as a lecturer. She completed her master's degree in 2000 in the field of agricultural microbiology from University of California. She continued her higher studies at University of Aberdeen and obtained PhD in agricultural microbiology in 2005. After returning to the department, she has been promoted to the capacity of the senior lecturer. She has been appointed as a professor in plant science in 2019. She is the current head of the Department of Plant Sciences, and she has authored 2 books and 2 book chapters, 40 journal articles in index journals, and more than 100 abstract publications. She supervised 40 undergraduates and 30 postgraduate research students. She is actively involved with Sri Lanka Accreditation Board for Conformity Assessment, Sri Lanka Standard Institution, Ministry of Agriculture, Ministry of Environment, Sri Lanka Council for Agricultural Research, etc. at various capacities. Now it's my pleasure to invite Professor Chandrika Nanayakara to deliver her plenary speech.
Thank you very much for the introduction given. Can I do the presentation myself? Please allow me to share the screen.
Madam,
Yes.
Madam, you can do the presentation now.
No, can I share the screen as well? I want to share the screen from this end. Is it possible?
Yes, madam, it is possible.
It's not giving me.
Madam,
Okay, I'll try. Thank you. Thank you so much. Right now, I'm Chandrika Nanayakara, attached to the Department of Plant Sciences. And good morning to you all. And thank you for the very nice introduction. I also would like to thank the organizing committee for giving me this opportunity to shed some light on the research directives in plant sciences. So why plant sciences are so important? Now, if you look at the basis of all life forms is plants. They make an indispensable part of human life as they provide food to sustain life, clothes to wear, medicines to treat ailments, and most importantly, the very air we breathe. So without plants, none of the higher organisms that are extant on this earth would not have been evolved. So research on plant sciences would invariably advance the quality of life and while achieving most of the sustainable development goals. So what exactly is plant science? So it is the science-based study of plants. The forms and interaction of plants are also involved. Insights into plant genome widen the horizons of classical plant sciences to embrace the studies on genes and gene interactions, plant microbe interactions, computational biology, bioinformatics, etc. And plant science is actually a multidisciplinary science having extended arms from chemistry, computer science, mathematics, microbiology, physics, etc. So the chemistry and plant sciences are having a long-standing history with respect to natural products. Plants and microorganisms are good sources of bioactive molecules having antimicrobial, antidiabetic, anti-cancer, then anti-quorum sensing molecules, etc., as well as there are molecules that we can extract to be used as natural dyes. So crude extracts with the antimicrobial activities have been detected in lower plants as well as in other components of the plant life. Now, Sri Lanka, we all know, is an island and it is blessed with a huge wealth of marine resources. Among those, algae have been investigated for biologically active molecules. Anti-inflammatory, antioxidant, cryoprotective activities, etc. have been well documented, and you can see some of the very important publications related to such products. And also, the macrofungi community in Sri Lanka have been used in indigenous medicine, and although it is not very prevalent among Sri Lankan, prevalent in Sri Lankan indigenous medicine, most of the countries are using heavily these macrofungi resources. And Sri Lanka is having a tropical climate and which is very, very conducive for the growth of large number of macrofungi. And these are also harboring very important bioactive molecules. So we conducted some investigations into dry zone macrofungi, and we have been able to reveal their potential to be used as sources of bioactive molecules. And also, novel bioactive compounds have also been synthesized and isolated, identified, and also synthesis in the labs has been conducted so far. And also, we have received a US patent. This is known to be the first US patent received to the University of Colombo, and this is with regard to anti-cancer compound extracted from a mushroom from Sri Lanka. And these mushrooms, in addition to their medicinal properties, wild mushrooms have been used in tropical cuisine. They also have therapeutic properties, and most of the indigenous knowledge about the uses of these wild mushrooms and being able to eat them, consume them, is disappearing fast. So therefore, the research attempts were conducted to identify these wild edible mushrooms and also to perfect the culture protocols. And we also conducted some of the nutritional aspect analysis of some of these wild edible mushrooms from Sri Lanka. And not only those, some of the plant chemistries make them a good source of natural dyes. The ability of natural dyes to color fabric and paper has been known since ancient times. Due to the non-toxicity, biodegradability, and antimicrobial properties of these plant extracts coming from various plant parts, we can use them to extract dyes. So some of the plants that we have used in developing such dyes is, you can see some of the dyes produced, although it is not being commercialized. We have the protocols developed. A red color dye has been isolated from Cisalpania sappan, which is known as patangi, and the dye is coming from the wood. And then there was another one where we extracted the indigo dye. This indigo dye is used in the coloration of denim, and a Sri Lankan plant, Nilavaria, which is scientifically known as Indigofera tinctoria, has been used in such studies and successfully isolated the dye as well. And one of the key pressing areas of research is in the area of bioremediation. This is one of the greatest environmental problems we face today. It is environmental pollution due to the mammoth volume of solid and liquid waste generated on a daily basis. Plants, fungi, and bacteria are key players in pollution abatement. Microbial solutions for waste treatment have drawn attention of researchers during the past few decades. So heterotrophic microorganisms are normally used in these studies because of their ability to produce a plethora of extracellular enzymes. These enzymes can break down most of the macromolecules. Actually, this is for their own survival. They try to get their food from the outer environment, as well as they try to make the environment habitable for themselves. So in that aspect, they try to either break down or sequester pollutants that are present in their immediate environment. So these abilities have been harnessed to control environmental pollution. So we have searched for efficient waste degraders and to treat polluted water with lipids and other food components using the microbial enzymes. as well as to degrade the plastic waste present in the environment. So not only the microorganisms, plants are also having certain capabilities, and we have conducted some research to use indoor foliage plants to remove air pollutants from indoor air with successful results. And then to advance the plant resources, the food resources especially, plant breeding programs are also in place. And plant breeding is in its simplest form is the application of genetic principles to produce plants that have more desirable characters. Although the plant breeding programs have delivered today's highly productive crop varieties, the rate of genetic improvement must be doubled to meet the projected future demands. So without new approaches that help boost productivity of staple crops through genetic improvement, global food security will be severely compromised in the next 2 to 3 decades. So understanding reproductive biology is the key step in breeding programs. So some of the important crops have been considered in our research agenda. One is shown here. This is, is a popular vegetable among Sri Lankan people, Tumbakaravilla, scientifically known as Momodica dioica. Here we identified that these are actually need to be hand pollinated because the natural pollinators are very, very, say, less abundant in the environment. So the research have been geared towards identification of natural pollinators have succeeded, but the problem is again the natural abundance to be very, very low. And then with that, we are in a program of characterizing black pepper varieties and probe for useful characteristics that would be needed in future breeding programs. So the wild varieties as well as cultivated varieties have been collected, and those have been analyzed for morphometric characters as well as the genetic characters. So this actually is still progressing, and we will be able to identify some important characters that can be utilized in the future breeding programs of piper. Because piper is an important crop which brings a large amount of foreign exchange to the country. And it is also known as king of spice, as you all know. So now the other problem is we can develop allied varieties, but maintaining this prototype nature is another obstacle in the plant breeding programs. So agriculture, in addition, the agriculture mechanization necessitates the synchronization of flower and seed setting of plants. So therefore, plant tissue culture-based micropropagation technique is an indispensable tool in this respect. Also, we are looking at the possibility of perfecting anther culture technologies. Anther is haploid in nature, and so whatever the characteristics present in a plant will be expressed in haploid plants. But haploid plants cannot be cultivated because they are sterile. So what we are trying to do is through anther culture to bring about the hidden characters, and then with various chemical treatments, you can make them a diploid so that some of the good recessive characteristics can be identified. And if that happens, those will be also important in the breeding programs. Apart from rice, this is about rice anther culture, and the regeneration of plants have also been done, and the protocols have been perfected. And apart from rice, we are also concentrating on tissue culture protocol development for micropropagation of crops like coconut, and then pepper, passiflora, etc. And also the molecular biologically, the transformed plants. Although in Sri Lanka, we still cannot go to the field with the genetically transformed plants. In the laboratories, development of protocols for genetic transformation is allowed. And so under that, also when the plants have been transformed, these tissue culture techniques are needed for the development of such transformed plants. And then this is kind of not really new area of biological sciences. And the Department of Plant Sciences actually is proud to say that this bioinformatics, this new field, has been introduced into the local university curricula. First, we did that in the Department of Plant Sciences. Now, the importance is that, metagenomic approaches are becoming increasingly popular in large-scale genomic applications. This allows the study of taxonomic and functional composition of microbial communities from environmental, agricultural, and clinical settings. Bioinformatics, the analysis using bioinformatic tools make it possible to mine huge metagenomic data sets and discover general patterns that govern the interaction between various components of the ecosystem. So it is a cross-cutting discipline which applies tools of computation and analysis to capture and interpret biological data, and that actually bridges computer science, mathematics, physics with the biological sciences. Right now, a wealth of genomic data is available to study plant genetic variation at molecular level that can revolutionize plant biology research. Anyway, the main components of bioinformatics are the development of software tools and algorithms, and then use of these software tools and algorithms to analyze and interpret biological data. So as we said, the special degree in bioinformatics have been introduced into our curricula, and with that, some of the research we have conducted have been highlighted here. And recently, we also completed metagenomic analysis of cattle milk microbiota in Sri Lanka, and the publications are yet to come. And that is the first of that kind of a study conducted in Sri Lanka. So as you can see, bioinformatics tools can be employed in mining for special genes and gene products, and also identify gene and protein regulatory networks, and then map them to the metabolomics as well. Then another research area we are having is plant microbe interactions. So the interactions could be of symbiotic in nature, where both parties, the plant and the microorganism, is benefiting, or they can be antagonistic in nature. Mainly, microorganisms are pathogenic on plants, causing various diseases and yield decline. So these plant microbe interactions are very, very complex, dynamic, and it's a continuous process and have a history that dates back to plant origin on Earth. These million-year-old associations have formed distinct assemblages that could either be beneficial or detrimental. And these have a great impact on agriculture production, as either the crop loss or yield increment could be the result of such an interaction. The beneficial interactions can be defined as some direct or indirect mechanisms, such as nutrient acquisition and transfer, nitrogen fixation, stimulation of growth through the production of phytohormones and other growth regulatory molecules. And also there could be antagonistic activities towards the plant, as I said earlier, and those, if you understand such antagonistic activities, then that can also be used to increase the crop productivity. So the severing plant microbe interaction is a critical component in recognizing the positive and negative impacts of microorganisms on plants. Understanding the plant biology of plant pathogens will help in designing control strategies, while microbe-microbe interactions will lead to identification of natural enemies as well. We conducted a research program to identify resistant genes for plant pathogens using chili as a crop, and an extensive survey on resistant genes in Sri Lankan cherry varieties. These varieties actually include the wild relatives as well as the cultivated varieties we are having. And the study revealed that none of the cultivated and wild relatives, both, are not to contain resistance to the disease that was under investigation, that was the anthracnose disease. So which means during the course of evolution, somehow the resistance had degraded, and which means still we have to look for some wild relatives, not basically chili varieties, but there are other surrounding varieties, and try to find out a way out. And also the natural enemies, as I told you, if we can look for natural enemies for certain plant pathogens, they have the potential to be used as biological control agents. So this is one instance where we have succeeded in that aspect. It has been reported that there's a new disease causing the flower rot of onions are present in some of the north and eastern areas of the country, the onion growing regions of the country. So this is affecting the inflorescence, reducing the seed set as well. So considered as a serious threat to the onion production. So investigations into these incidents led to the identification of this pathogen complex. We identified it is known as onion black mold complex. And then from the same areas, we have been able to identify some antagonistic trichoderma species. Using these species, now you can see the plate assays have been used to identify their pathogenicity, the biological control activity, and then field experiments have been conducted, and simple formulation have been developed as well. Now this has been now tested by the farmers in those areas. So this is another incidence. Actually, this is still progressing where endophytic microorganisms. So endophytic microorganisms are the microorganisms that are living inside the plant bodies, but those are not pathogenic microorganisms. They have some growth promoting activities there. So we are now probing into endophytic microorganisms and try to identify their activities against pathogenic microorganisms. So this search for biological control agents, here in this case it is for rice, and so anyway these are green solutions that can lead to the reduced use of toxic agrochemicals. And I think under the current situation where agrochemical usage has been limited or stopped in the agriculture sector, these research can be of use to the government to develop green solutions. And so this is another success story. So advancing expertise in plant interactions with symbiotic microorganisms to create resilient crops and improving management of agriculture systems is one of our major research areas. Microbes. Here again, we are using endophytic microorganisms, and this particular endophyte has the ability to fix nitrogen within the plant body. Protocols have been developed to introduce the organism into the rice plants, and then this nitrogen fixation, the organism will be conducted within the plant, as I said earlier. So this endophytic nitrogen fixer introduced into the rice plants led to 50% reduction in urea application for rice crop. And currently, we are in the process of developing nitrogen-fixing microbial consortium to satisfy the total nitrogen demand of rice. And so these are some of the publications stem from those research. And similarly, we have research conducted trying to value add a polar rock phosphate, which is a natural phosphate fertilizer, but due to its low solubility, it is not recommended for short-term crops. And here we are trying to identify microbial solution to increase the solubility of these rock phosphates. And again, the very first experiments conducted were on cutting down TSP application in rice crop system. And the pot and field experiments conducted have been able to cut down 50% of the TSP applied, and further improvements are in order to a complete replacement. So the same technology has now been extended not only to the rice, but also to some other important crops. So here the technology is, you can see the pot experiments have been completed, and we have taken some data from the field experiment site as well with respect to cinnamon. And then the same thing has been extended to crops like pepper and chili. So these also gave us promising results, and therefore the next step is to develop a phosphate bar fertilizer using the identified efficient phosphate solubilizing microorganisms. And then we are also into food industry. Again, the use of microorganisms in the food industry. You know, the microorganisms are the key players in food spoilage, but at the same time, most of them are used in developing new flavors and new foods. So they play an important role in food industry, especially in the fermented food industry, as well as they are used as probiotics or to produce various probiotic foods. Various microorganisms have been used as starter cultures to produce fermented dairy foods, such as cheese, yogurt, curd, etc. And this fermented food production is relying heavily on starter cultures. If you look at the global scenario, the global starter culture market was valued at USD 1 billion in 2020. And it's projected to reach USD 1.3 billion by 2025. So this is a flourishing area. And there is a growing demand for dairy and dairy-based food in all the countries of the world. And there is an increase in the consumption of probiotic foods. That actually came with the knowledge that these microorganisms, once you ingest them, they can colonize your colon and the small intestine and bring about a plethora of beneficial effects. So healthy gut is actually meaning a healthy lifestyle. So therefore, the demand on the probiotic foods, new flavors, etc., is on the increase. So therefore, we are now looking at as I told you, we completed a metagenomic analysis, and then that is a culture-independent method of analyzing the milk microbiota. At the same time, we use culture-dependent methods, so normal plating techniques, and the microorganisms that are grown on the cultures have been isolated, and genotypic identification has been done. Out of hundreds of different microorganisms yielded from these culture techniques, we have identified about 40 different yeast and lactic acid bacterial species that are having some nutritional applications and that are having technological applications in industry and also having the potential to be developed as probiotic foods. So analysis towards this development of probiotics have been completed as well. So now we have a good collection of such microorganisms at ITI, because this is a collaborative research conducted with ITI, and we have a collection of probiotic lactic acid bacteria and yeast culture collection with us right now. And we are now trying to see the applications in various food industries. And also some aspects, as I told you, spoilage is another problem coming from the microbial in the growth on food. You know, as an island, we are having a rich supply of fish and seafood. And Sri Lanka has been reputed as a quality fish and seafood exporter. We supply predominantly yellowfin and big eye tuna species to international markets. Faults in the process chain may introduce harmful microorganisms into fish, rendering them unsuitable for consumption. And some of the complaints have been received from the export companies with regard to the histamine formation in yellowfin tuna. So if you consume fish containing this histamine, it can cause various diseases, especially allergies in humans. What happens there is bacteria can produce histamine from histidine present in the fish. Then what we did was we looked at the process chain, and then we identified the critical control points where these introduction of histamine forming bacteria can contact the fish. And then after identification of these control points, now we have given the recommendations to minimize or say to control the introduction of such organisms into yellowfin tuna. Right. Now, another priority research area is the studies on biodiversity. You know, the variety of life forms, the ecosystems they form, and the habitats in which they live can be included under one term, that is biodiversity. So normally we explain biodiversity under three themes. Those are the species diversity, genetic diversity, and ecosystem diversity. And it is important to learn about these diversities because habitat loss from exploitation of resources, agricultural conversions, and urbanizations. Those are the largest factors contributing to the loss of biodiversity. And then ecosystem degradation, introduction of alien species, and climate change are already seriously impacting the Sri Lankan biodiversity. Therefore, assessing the biodiversity, documenting the biological wealth, modeling the impact of such threats on biodiversity, control of alien flora and fauna, etc., will enable the development of appropriate conservation and sustainable development strategies. Plant and microbe diversity are our priorities in biodiversity research. So plant diversity studies lead to the new taxonomic delineations, and also macrofungi research revealed some new records of mushrooms in Sri Lanka. So these are actually some of the milestones in biodiversity research. So these are some of the collections we had during our field excursions into the dry zone forest of Sri Lanka. All these are actually part of the microflora that we have collected from those areas. Altogether, we can see a large number of macrofungi have been documented, and most of them are actually, I think, there are some new records, and the new records have been published as well in reputed journals. So all these are new records, and now we have been able to print a coffee table book, including all these nice photographs, and any mushroom enthusiast kind use this as a field guide for the identification of mushrooms. Then urban ecology is actually another priority area. The rapid expansion of cities and urban population necessitates the study of interaction between living organisms and urban environment. So that comes under the purview of urban ecology. So it is defined as the environment surrounded by man-made structures, such as residential and commercial buildings, paved surfaces, etc. So urban ecological studies, urban ecology studies the ecological processes in urban environments. So those are including all aspects of the ecology of an organism found in an urban area, as well as large-scale consideration of the ecological sustainability of the cities. And some of these photographs, actually, you can see these are coming from the near the perimeter. This is the perimeter of the university, where the disturbances to the plants have brought them down. And you can see here in the University of Colombo, there are a large number of trees have fallen down due to the constructions, as well as due to the invasion by the species that are not really needed to be there. So they are killing our biodiversity in the Metropolitan University as well. Floriculture is another important area to Sri Lanka. Sri Lanka presently accounts for less than 0.2% of the global flower trade and has the potential to secure much larger share. The floriculture trade in the country has risen to over 1.5 billion rupees annually. And we are exporting cut flowers, cut foliage, live plants, bulb forms, tubers, etc. But the domestic trade is also there, which is primarily comprises of cut flowers and indoor ornamental plants. So the major problems associated with cut flower industry is lengthening the vast life of cut flowers and foliage. Especially plant foods and ethylene inhibitors or scavengers are in constant demand as a remedy for this problem. So researchers in the Department of Plant Sciences have been able to develop a plant food recipe to lengthen the vast life of gerbera. So the recipe is here, and now actually the researcher is involved in disseminating this knowledge to the Vidata centers, to the flower growers and the other people who are involved in cut flower industry. And also there's a huge demand on preserving the fresh flowers. Instead of using our flowers, people are ready to use the preserved flowers, especially the wedding bouquets are preserved. Some other foliage and flowers are used in costume jewelry. So the color preservation as well as texture preservation is there in the demand. So natural color preservation studies have also been conducted together with the texture preservation. So some of the treatments and the results you can see here, and a protocol to preserve flower texture has been developed for the roses, and you can see different treatments to retain the color as well as texture of the fresh flowers. They are shown here. And then the plants and the environment is also an important area. Being unable to move, plant growth and geographic distribution are greatly affected by the environment. So if we can recognize the roles of these factors, we will be able to diagnose plant problems caused by the environmental stressors. And also if any environmental factor is less than ideal, and in order to survive, plants need to adapt themselves to such conditions. So these adaptations are manifested through morphological, anatomical, and genetical responses. So studying such adaptations enable us to understand plant responses through modeling, and information derived can be used in predicting plant responses to environmental stress. So this will also shed light on crop responses to climate change, and predictions will help in keep selecting or developing climate smart crops. So plant sciences. Now, as you can see, most of the plant science research are not rocket science. Most of them are preliminary and at basic level, but the outcomes are having significant impact on the human life. So, some of the publications on plant and environmental interactions are also shown here. As I have already said, so plant sciences are offering numerous research opportunities. And here, actually, I have presented the surface only. Being an interdisciplinary science, we can work with experts in any other discipline. So I think as when you are going through the presentation, listening to my presentation, you must have find some research areas that are of interest to you, or you may have developed some ideas of collaborative research as well. So the Department of Plant Sciences will be more than happy to embrace such opportunities, such collaborative work with any other scientists in any other field. So with that, I would like to wind up my presentation, and thank you very much for your attention.
Thank you very much, Madam, for that informative speech. Now it's the time to present token of our appreciation to Professor Chandrika Nanayakara.
Thank you.
Thank you very much, Madam. To introduce our next speaker, Senior Professor Devaka Veerakorn, I would like to invite Professor Anjana UK Piyadasa, the Acting Dean, Faculty of Technology, and also he's the head of the Department of Environmental Technology.
Okay, thank you very much. I'm happy to read the citation for Senior Professor Devaka Veerakorn. Senior Professor Devaka Veerakorn graduated from the University of Colombo in 1985 with a major in zoology. He joined the Department of Zoology, University of Colombo in 1986 and received the Fulbright Scholarship in 1988 to the Illinois State University, USA, where he obtained his MSc and PhD. In 1995, he returned to Colombo University, where he currently holds the position of the senior professor. His main research interest in the conservation of threatened species, especially Asian elephants in Sri Lanka. He also serves as the chief technical advisor on fauna for the red listing program of Sri Lanka from 1996 to date. He is also technical advisor to the Ministry of Environment or Conservation of the Threatened Species. Senior Professor Devaka Veerakorn has also served as a consultant on biodiversity to IUCN, Asian Regional Office, as well as IUCN Sri Lanka. He is member of the Asian Elephant Specialist Group. Thank you very much. And Senior Professor Devaka Veerakorn, over to you.
Good morning, ladies and gentlemen, Madam Vice Chancellor, Dean of the Faculty of Technology, distinguished guests. This is our planet. Planet Earth is a very old planet, roughly about 4.5 billion years old. And the historical time period of our planet can be divided into eons, eras, periods, epochs and ages. Now, October 4, 1957 marks the beginning of the space age, and this is what eventually put a man on the moon. And Neil Armstrong, that man who walked on the moon, said, "One small step for man, one giant leap for mankind." Indeed, ladies and gentlemen, we have grown in leaps and bounds. But I'm not going to be talking about that because I'm no rocket scientist. I'm a simple ecologist, and my main interest is conserving species. And therefore, the focus of my presentation today is use of technology for wildlife conservation. What I would like to point out is how has technology revolutionized the discipline of wildlife conservation during my brief presentation. First of all, I think we should explore the question, why do we have to conserve wildlife in the first place? Because what I'm showing you here is a graph which shows how the number of species on our planet has changed over time. 600 million years ago, an event that took place on the planet, which is referred to as the Cambrian explosion, showed that the life on planet started expanding in an unprecedented rate. And you can see life begins to increase, the diversity begins to increase, and then you see a drop here, again an increase, again a drop, and so on. So basically, if you look at the last 600 million years, you can see there were periods where there was a great deal of speciation, new species arriving on our planet, and there were periods where species begin to disappear at an alarming rate. And these periods are referred to as mass extinctions. Up to now, we have had 5 mass extinctions on our planet. And there is enough data to show that we are currently living through the 6th mass extinction. Now, the previous mass extinctions, we had nothing to do with it because we were not there on the planet. We only appeared about 200,000 years ago. However, the current mass extinction is primarily driven by human action. What I'm showing you here is a concept that is referred to as planetary boundary concept, where they have identified 9 boundaries that we should not cross if we wish to ensure that life on the planet can sustain without any trouble. However, when you look at this graph, you can see we have already exceeded the boundary at least on 3 out of the 9 categories that we're talking about. And in 2 of them, we have exceeded it by a significant amount. One of these is the biodiversity loss in the planet. We live in an era where there is unprecedented loss of biodiversity, mostly due to human activities that are taking place on the planet. And this is why we feel that conservation of wildlife is necessary to slow down the rate of extinction of species, because we do depend on these species for the services and goods they provide, and their loss will eventually have a huge impact on humankind as well. Now, how do we ensure wildlife conservation? So the approach to conservation deals with dealing with the drivers that have resulted in loss of species. Now, these drivers can be broadly categorized into habitat-related drivers, which includes loss of habitat, habitat conversion, habitat fragmentation, habitat degradation, and so on. Then there are species-related drivers, which includes overexploitation, illegal wildlife trade, human-wildlife conflicts, introduction of invasive species, which has led to loss of biodiversity in many parts of the world. And on top of all this, we have climate change taking place, which is exacerbating the existing drivers even further. So we live in a challenging time where we have to deal with all these issues. Let me briefly then explain how technology or technological innovations have helped wildlife conservation. Because I'm showing you here 3 major technologies that have influenced wildlife conservation in a very, very significant manner. One of these is space-derived data usage, or using satellites to look at the surface of the planet as well as to monitor movement of species. Then the second major technological innovation is drone technology, which has given us the ability to approach even very inhospitable places or places that humans cannot easily reach, or look at situations without disturbing species. And then the camera trap technology, which allows us to place a video camera or a camera in a remote location, and the camera will take various photographs which will help us to figure out what's happening in that part of the world. So these technologies have revolutionized the way we do conservation, and I'm going to just take 5 examples, and all from work that has been done in Sri Lanka, mostly the work that I have been doing over the last few decades, to explain how these technologies have helped us to resolve certain conservation problems. So the 5 case studies I'm going to present is, one is about monitoring habitats. How can we use technology to monitor habitats? How can we use technology to identify suitable habitats for species? How can we reduce the impact of development using this kind of technology? And the 4th example is, how do we use this technology to monitor migratory species? And then finally, how can we use this type of technology to reduce human-wildlife conflict, especially species that are involved in conflict, which are also globally threatened, and therefore conflict is pushing them further and further towards brink of extinction. So let's first of all look at how we can use technology to monitor habitats. One of the things we can use, one of the uses of technology is to monitor what is happening to habitats. What you're seeing here is the area that is referred to as Wilpattu National Park, area north of Wilpattu National Park, and this is a satellite image that has been taken before 2009. And you can see this entire area is covered with forest. And then you can see this is a settlement that created a great deal of social unrest in Sri Lanka, the resettlement of people north of Wilpattu, which led to a massive environmental movement. And you can see why when you look at these 2 pictures. This picture shows what has happened to the forest around here. And this is the new settlement that has been established north of Wilpattu, which has resulted in loss of significant amount of habitat. Then secondly, we can use satellite imagery to monitor habitat conversion. We lose about 7,000 hectares of forest like this every year because they are being converted to either reservoirs or agricultural land or human settlements. So once again, satellites gives us a way to monitor how habitats are being converted and the consequence of it. Then satellites based data helps us to also understand the concept of habitat fragmentation. The next set of pictures I'm going to show is from Vavania. This is a satellite image taken in 2006, and you can see this entire area is covered with forest. And during this time, you all remember we had armed conflict happening in the northern region, which was culminated in 2009. Now, after the culmination of the armed conflict, there was a massive move towards development. And this is what development looks like. And you can see this forest is now going to be fragmented into small pieces due to development of roads, resettlements, agriculture practices, and etc. So this sort of satellite-based tracking helps us to see what is happening to our habitats. Of course, ideally, we should be able to use this data to influence further loss of habitat or further fragmentation of habitat, but at least it gives us a handle to quantify what is happening to habitats in the country. And also, this kind of technology can be used to identify suitable habitats. This is a project that we did again close to Wilpattu. This is the Kalaoya River Basin. What we wanted to see is we wanted to identify habitat availability for some of the critical endemic and threatened species. So once again, satellite imagery along with GIS technology allows us to predict what are the ideal habitats for a given species by plotting some of the available distribution data, and this particular software will then will identify habitat parameters and tell us what are the ideal habitats for the species, so we can make a judgment as to how much habitat is available and therefore how much population we can maintain. And also when we are doing studies, this kind of predictions helps us to narrow down our search patterns so that it gives us a better handle of where to look for if we're looking for these species. And then the third example I'm going to show you is how we can use this kind of technology to reduce the impact of development, because development has a significant impact on wildlife habitats and species. So now it has been accepted in Sri Lanka that development should be associated with wildlife management plans and plans to ensure that the impact of development can be curtailed as much as possible. So this is a project that I was involved in the northwestern province of Sri Lanka. What you see here is a major canal project that is taking place in the northwestern province. And this canal project will provide water for more than 600 tanks, and therefore it's going to change the cropping intensity and cropping patterns in the northwestern province. And this is the Galgamua Mahava area. So we were asked to go and see what would be the impact of this on habitats and to identify those habitats that we have to conserve in order to ensure that this development is sustainable. So once again, we turn to satellite technology, we use satellite imagery and available data. And by using modeling, we have identified these red areas are the areas that are critical habitats that needs to be set aside for conservation so that the development can take place without really compromising our critical wildlife resources. So this kind of studies will lead to identify how we can do development without seriously compromising our wildlife resources. This is another example I'm going to show from, again, this is one of the largest reservoirs that have been established in recent time. This is a picture of the Muraga Kanda reservoir. And when you build a reservoir like that, first you build the dam, and then once the dam is completed, you start filling the reservoir. So what you see here is what happens, how the reservoir is being filled. It's a time series that you can see here. You can see the reservoir is filled about 25%, about 40%, and here about 50%. So you can see a gradual increase in the filling. What I want you to focus on these areas that I have marked as A, B, and C. So when you fill a reservoir, the bed is not flat. The bed has an undulating topography. So water starts filling in the low-lying areas, and the higher grounds gets inundated later. So when the water, when you start filling it, you can see in this area, this is a higher ground, this is a higher ground, this is a higher ground. Water is filling around it, and you can see at this point, all three locations are connected to the land. And if you move to this, you can see this peninsula now gets completely encircled by water. So you create an island here. This is still remaining as a peninsula, and this region also has now become an island. And when you move on to this, you can see the island has become smaller. This peninsula has turned into an island, and this island has disappeared. So when you start filling this kind of reservoir, the animals, their general tendency is they are used to floods. So when a flood comes, animals tend to move to higher ground. So basically what happens is animals will start moving into higher areas. However, what you see here is not a flood. A flood will come and recede. Whereas when you start filling a reservoir, the reservoir level begin to increase, and the animals who are trapped in higher ground will eventually become inundated. So when you fill a reservoir like this, we have to have an extensive rescue program to rescue animals who get stranded in the higher grounds. So this is another project that we have done for the Mahaweli Authority, where we have used satellite technology, and this is again the Morogahkanda Reservoir, and we've used satellite technology to identify the topography of the reservoir, and we ran a model which shows how the islands are formed, or where the islands are formed, and also at what level of filling. So you can see here in this table shows when you fill the reservoir up to 149.5 MSL, these two islands A and B will appear. And when you increase the filling, island C will appear. And likewise, some of these islands will appear when the water level reaches a certain height, and then as the water level keeps increasing, these islands will disappear. So this kind of modeling will help us to identify where the islands will be formed. Otherwise, we'll have to be searching this entire large reservoir, and it takes a long time to search the area to find where the animals are stranded. But with this kind of information, they know by looking at the water level, where the islands will be, so they can directly go to these islands and start rescuing animals that are stranded, which will make the rescue operation very efficient and cost effective. And also we will be able to rescue most of the species using this kind of technology. And also we can use the satellite technology to monitor migratory species. This is again a project that is currently being done by Professor Sampath Siniviratna of Department of Zoology and his PhD student. They're using satellite based tracking to see how the migratory species behave. Where do they come from? Where do they go? And in between, where do they land? So that we can migratory species conservation transcends beyond the boundary of a country, because these birds migrate through multiple countries. And if you look at this particular example, they come from Siberia, cross over the Himalayas. You can see them crossing over the Himalayas here, then come through India all the way, and their final resting place is Sri Lanka. So during northern winter, they come to Sri Lanka, and then when the summer comes in the north, they go back. But this kind of tracking helps us to identify what is the specific route they take, where do they stop, so that will tell us those are the places that we have to protect, because in between they'll have to stop and refuel, and if we destroy those habitats, they will not be able to complete this migratory route. So basically, this is again to show one example as to how satellite technology can help us to do conservation in a more informed manner. And finally, I'm going to be focusing on how we can use this modern technology to reduce human-wildlife conflict. In Sri Lanka, we have human-wildlife conflict, which is a very critical conservation problem, because most of the species that are involved in conflict also happens to be globally endangered species. And because of the conflict, these animals are killed at an alarming rate. And as a result, these species become pushed more and more towards the brink of extinction. And therefore, it is important for us to understand how conflict happens, why conflict happens, and what kind of measures can we introduce to minimize conflict so we can ensure long-term survival of species such as this. And incidentally, these two species are found only in Sri Lanka, and the Asian elephant is a symbolic animal in Sri Lanka with a long history, and it's also economic resource because majority of the tourists who come here are here to see elephants in the wild. However, we lose nearly 400 elephants annually, and which is a very, very high death rate when you look at a very small population that is found in Sri Lanka. So over the last 20 years, much of my research focused on trying to understand what is causing conflict and how can we influence this process, bring solutions that can mitigate conflict, which will ensure a long-term survival of the species. So we started work in 1995, where we started tracking animals to understand how does a conflict animal behave. So this is a fairly involved process. What we used to do at that point of time is using radio collars. So you have to tranquilize the animal, and once the animal comes down, you have to place the radio collar and revive the animal and then track the animal. And this is what you can see is a radio-collared animal in Yala. So basically what we do is that the animal is fitted with a collar which has a radio transducing device which emits a radio signal which you can pick with an antenna like this. So basically what we do is we pick the radio signal from 3 locations, a process called triangulation, and then we try to identify where the animal is. So once you estimate the location, you have to go by foot and make a visual confirmation, which is a fairly dangerous thing, because in the forest, sometimes while you are looking for the animal of your interest, you may run into other elephants. But this is the kind of technology we used during our early studies between 1995 and roughly up to about 2005. After that, this satellite technology helped us to come up with better collars that can be monitored from satellites instead of having to monitor them using antennas and homing them. We can basically track where our animal is by using a satellite collar, where the collar contains a GPS unit and it emits the location or the coordinate of the animal into a satellite, and via satellite we can download it to a computer, and then we can map it and the coordinate, and we know exactly where the animal is. So this system can be set up for the animal to send a signal every 1 hour or every 4 hours, or whatever the frequency that we desire. When we increase the frequency, the battery life goes down. So it is a trade-off between how much information we need and how longer we want to use the collar. But this kind of technology makes things very easy, because when we were using this kind of technology, the whole day we can maybe track about 1 elephant. And I had like 8 elephants that I was tracking. So I have to use the whole week to get just 1 data point per animal. Whereas with this technology, now I can get maybe 1 data point every 4 hours. So it increases the amount of data we can get. It increases the resolution of the data, and therefore we can reach better decisions. So this is an animal fitted with a satellite collar. You can see this is the satellite transceiver, and we put a weight here to ensure that the GPS unit is sitting on the top, so it can emit the information to a satellite without disruption. And this is one of the studies we have been doing in Galgamo area. What you can see, this each color indicates a herd of elephants. And you can see, if you look at this, there are about 7 herds, and we are tracking, each herd has an animal with a satellite collar, and we are tracking their movements. When you look at this picture, you can see the animals extensively use this area. This is Mahagalgamo tank, and this is a forested area. So you can see that they use this area extensively, and they also use this area. And they keep crossing from this particular location. They don't move all over the place. They are specifically using this corridor to cross between these 2 habitat patches. Now, if we try to use visual observation, it will be impossible to, near impossible to identify this kind of a corridor. But this type of technology helps us to identify this corridor, and therefore by protecting this corridor, we can ensure that these elephants can move around without really coming into human use areas. These are all human use areas, so we can protect the human use area and ensure that the elephants can continue to move between their habitats without coming into conflict with people. This is another study that we have done again in that area. What I'm showing you is a time series. I said we can extract a data point every 4 hours. So we have collected data for a long period, and then we have grouped them by time. So what you see here, this yellow area is a paddy cultivated area. This green area is a forest patch, and these black dots indicate where the elephant with the collar is found at that particular time. So this is where the elephant used to be during 12 noon, during the period of monitoring. And you can see at 12 noon, most of the data indicate that the animal is staying inside the forest patch. Except for this, all the other data points are within forest. And then this is at 4 p.m. You can see at 4 p.m. all the data indicates that the animal is moving towards the periphery of the forest. And by 8 p.m. you can see they're out here in the paddy fields raiding the paddy cultivation. And by midnight they have gone deeper into the paddy areas. And by 4 a.m. you can see they're coming back to the forest patch. So this again helps us to understand how crop raiders behave. They hide during daytime, raid at night, sort of like Jekyll and Hyde kind of scenario. But by this type of technology, we can easily visualize the crop raiding behavior of the animal and can come up with ways as to how we can tackle this problem. And ideally, what we should do is, if we have a developed area like this, we shouldn't have forest patches like this in the middle, because this helps the animal to hide during daytime and attack. So this again helps us to identify what is the land use planning approach we should have. We should have human use areas separately, forested areas separately. But if you try to have a mix of it, it can always lead to higher level of conflict. And you can see by 8 a.m. they are back in the forest. And in order to address the human-elephant conflict, one of the most effective solutions we have is this type of electric fences. So electric fences are meant to keep elephants in the forest and to stop them from coming to human use areas. And because when they come to human use areas, that invariably leads to conflict. So once you put up electric fence, you have to maintain it to ensure that it works efficiently all the time. And if you do not maintain it, it can lead to a situation where the elephant will breach the fence and come out to the human use areas. But what we see in Sri Lanka, we have large amount of thousands and thousands of kilometers of electric fences. But when we study the efficiency of these fences, we see many of these fences are not located in the right ecological boundaries. As a result, they have become ineffective. Many of them are not properly maintained. So even though fences are fairly a good solution to the problem, most of the fences we see are not providing the desired result. And also to relocate the fence is quite cumbersome, because you have to bring down the fence and then redo it, which is very labor intensive and cost. It was a fairly heavy cost. So one of my PhD students is currently trying to develop a different kind of a solution, which again involves electricity. We borrowed this idea from dog electric shock collars. You can see here a dog wearing an electric shock collar, and this kind of collars are used for training dogs. So basically what happens is you define a certain point that the dog cannot cross, and as the dog starts moving towards that virtual boundary, it first emits a noise warning, and if it continues to move, it gives a shock. And if it doesn't turn around, it gives a shock at a higher frequency until the animal turns around. And this has been shown to be very effective, because only the first time the animal will need to be shocked. And after that, the animal will learn to associate the noise with the shock. So the moment you give the warning signal, it turns around. So this technology is also used by cattle farmers by developing electric collars that can help them to move their cattle. So these days, the cattle farmers don't have to get on a horse and move their cattle around, whereas they can place a collar like this, and using an iPad, they can control their movement. They can place a virtual boundary, and the animal will only operate within this. If they try to cross it, they get a shock, and therefore they will turn back. And if you want to move them around, you can redefine your boundary. So you can move them from one patch. When the patch is overused, you can move them to a different patch, allowing that patch to regrow. So this way, the farmer is capable of controlling the movement of the animal. And if it wants to weigh the animal, they can also use this technology to direct them onto a weighbridge. So when the animal moves through the weighbridge, the farmer knows exactly how many kilograms of cattle he has. So this has revolutionized the cattle farming. This particular example is from Australia. So what we are trying to do is we are trying to see whether we can develop a similar collar so that we can control the movement of elephants, not cattle, but because conflict-causing elephants, a lot of times if we can prevent them from going to human use areas, we can reduce the conflict tremendously. So we are working on a collar that can be fitted into an animal, works in the same way, and using iPad, we can define what are the areas that it can use and what are the areas that it cannot go to, and thereby we can prevent them from going into human use areas, or we can selectively move them around so that there is no overgrazing and so on. So, so far this is at a very experimental stage, but we have seen some very positive results, and we are hoping that if this technology works out, this would give us a very strong way to deal with human-elephant conflict. So in conclusion, what I can say is space-derived data have helped us a great deal to ask questions and get very defined answers. However, I would also like to point out that space-derived data does not serve as an alternative to traditional approaches that the conservation biologists use. I'm showing you this diagram. You might recognize this gentleman is Professor Sarat Kotagama, and that was me many decades ago. And this photograph was taken when we tried to climb the highest peak in Singaraja. So this expedition took us 4 days to reach the peak. And most of the time I felt that we were walking in circles, because at that time all what we had is a compass and a map to find our way. But today we have GPS units which we can carry with us, which can help us to find the way much easily. If we had a GPS, we would have done this in 1 day. So what this shows is technology will help us to do things faster, more efficiently, but technology won't help us to climb the mountain. Whatever happens, we have to physically go and get on top of that mountain. So that's why I said technology is not an alternative, but it provides us a powerful tool to get better answers to all questions. But for a conservation biologist like me, it's very helpful, but I leave you with the note, conservation biologists always work out there, because as God would say, world is my laboratory. Thank you, ladies and gentlemen. And I would like to thank Faculty of Technology for giving me this opportunity to come and talk to you. Thank you.
Thank you very much, sir, for that insightful speech. Now it's the time to present token of our appreciation to Senior Professor Devaka Veerakorn. Thank you. Ladies and gentlemen, now we are at the end of the plenary session. Thank you very much for joining with us. We move on the agenda. After 5 minutes short break, we will start the technical sessions of the 5th Annual Research Symposium of the Faculty of Technology. All the participants join via the online and physically. Please stay tuned. is to begin the technical sessions. To chair the technical session 1, I would like to call Dr. Jayani Vevalvala and Dr. Kanchana Abesekara from the Department of Agricultural Technology.
Thank you, Dr. Hansika. Good afternoon to you all. Welcome you all to the technical sessions of the Annual Research Symposium, Faculty of Technology, University of Colombo, 2021. So as Dr. Hansika mentioned, this is the session number 1 of the technical sessions of the Annual Research Symposium. And this session is chaired by myself and Dr. Jayani Vevalvala. So first of all, let me inform the guidelines to the presenters. Each presenter has 10 minutes for the presentation, followed by 5 minutes for answering the questions. The recorded presentations will be played during the sessions as we are conducting the sessions online. However, the presenter should present online for answering the questions raised by the audience. So without taking much time, let me introduce the first presenter today. The title of the presentation is Assessment of Energy Consumption and Reduction Techniques in Black Tea Manufacturing Process of Law Country Sri Lanka. So authors of this paper, M.D. N.D. Pereira, G.Y. Jayasinghe, S.P. Navaratna, C.M. Navaratna, G.D.C.P. Galpaya, and G.A.U. Jayasekara. The paper is presented by Ms. M.D. N.D. Pereira. So over to you, Ms. Pereira, to present your research paper.
Good afternoon. I'm Nimeshika Pereira from the Department of Agricultural Technology. The study was conducted on assessment of energy consumption and reduction techniques in black tea manufacturing process of Low Country Sri Lanka. Here is the flow of our presentation. The brand of Ceylon tea, a quality production from Sri Lanka, as we all know. With this greatest and famous brand symbol, we are in the 4th place among tea-producing countries, while being 3rd at the world export market. In both tea-producing and tea exporting, China and India in 1st and 2nd places. But our delicious flavor and smell, we are in the 1st place of orthodox black tea supplying to the world market under the Ceylon tea symbol of quality. When considering about the black tea production by elevation in Sri Lanka, the highest contribution has been given by the low-grown factories. So in here, the graph indicates that during the last decade, low country contribution has been continued with around 62 to 63% from the total production, while making a big gap with other 2 regions. But according to the FAO reports, the highest cost of production is recorded from Sri Lanka. We are in the 4th place of the production, we are in the 3rd place of exportation, but we are in the 1st place of cost of production. So, in detailed figures of cost of production at the last decade, it has been continuously increased year by year, which was highest at the last year of 2020. And it was around 600 rupees per 1 kilogram of made tea. When we compare the situations of India and Sri Lanka tea industry, here we can see the energy cost per ton is higher than the material cost in Sri Lanka. But in case of India, their raw material cost per ton is less than the energy cost. and also the energy cost in Sri Lanka is higher than the India. When we consider about the energy use in the different stages of tea life cycle in 3 tea growing regions, and normally in case of tea life cycle, the highest energy consumption has been recorded at the use because of water boiling, but after that the highest energy consumption has been recorded at the processing. When we consider about the 3 tea growing elevations in Sri Lanka, in case of low elevation, the producing energy consumption is higher than the other 2 elevations. With these criticisms, our study was targeted to analyze the energy consumption in low country tea factories with these objectives. First one was to assess energy consumption in low country tea factories and then analyze specific energy consumption. It means the amount of kilowatt hours per kilogram of made tea, and then to evaluate electrical energy use at different steps of black tea production, and finally to identify strategies for saving electrical energy in tea factories. In materials and methods, this is the study area. It means low country tea factories. All 3 tea factories were selected from the low country. Under these criteria, it means elevation was below 600 meters and average annual rainfall and temperature were 2500 millimeter and 27 degrees consequently, and then annual average production was especially concerned. In 3 tea factories, it was more than 3 million kilogram per year, and also again we concerned about the customer category in electricity bill. It was I2 and metered at 420-30 volt nominal and where the contract demand is less than 1000 kVA. These are the factors that we consider for purposive sampling of our 3 tea factories. In data collection, especially electricity bills and production data were concerned. And in case of electricity bill, electricity consumption was consumption data was gathered for 3 phases: peak, off-peak, and day. And apparent power, active and reactive power was concerned. And also production data were collected according to the made tea, refuse tea, and fresh leaves per month average. And these data were collected for 36 months. And also we collected some secondary data from Central Environmental Sustainable Energy Authority and previous studies. From the Sustainable Energy Authority, we get energy benchmarks for the low country tea factories. Here is the process of black tea production. Especially Sri Lanka is the highest orthodox production. It means black tea can be produced from 2 ways, orthodox and CTC, but in Sri Lanka highestly we produce tea from orthodox. Here is the process, and here is the electricity and thermal energy use at each step of made tea production. In results and discussion, first we assess the energy consumption in low country tea factories. Here are the 3 graphs that indicates the 3 factories electricity consumption. According to that, we can clearly see the day consumption is the highest at all 3 tea factories, and it is more than 50% of the tea process. It means tea process carried out in the daytime. And also this is the least payment from another 3 areas. So here is the electricity use in the factories according to the 3 phases: peak, day, and off-peak. According to that, we can see in 3 factories, all 3 factories, day consumption is around 50% and off-peak consumption is around 30%, and the peak consumption is the least. It is the best practice that factory can be done. So here is the specific energy consumption in each factory. This was calculated by taking electricity use per average month and total made tea per and also average made tea. By using those two parameters, we calculated specific energy consumption, and it was least at the factory 3, and it was 0.74. And when considering about the energy benchmarks which was given by the Sustainable Energy Authority, it was 0.82 kilowatt per 1 kilogram of made tea for low country, and it has been successfully achieved only one country, only one factory. It was factory 3. When we consider about the electrical energy use at different steps in here, we use energy detecting meters or the electrical energy consumption meters. We use especially EPRO 1000 meter for this one, and we place these meters for the motors of these each steps of these processes in two factories, especially in factory 1 and factory 3. We analyzed that withering troughs has used the highest percentage of energy from all process. In case of factory 3, these troughs, withering trough usages are less than the factory 1. When compared to the previous studies, the highest energy consumption has been reported in both factories as the previous studies in withering troughs. And also according to the withering, if we are doing a correct and instant significance withering for the black tea production, then we can reduce the cost of production also. In our study also, we can prove that by reducing the energy, we can reduce the cost of production also. Here, the objective 4, strategies for saving electrical energy. Here is the factory 3. They have used especially variable speed drives and variable frequency drives, VFDs, and capacitor banks for their energy saving, and also only one high efficiency motor to the withering trough. Here is the withering trough, and they have used these VSDs, VFDs, and capacitor banks to save their energy. It means to, in case of apparent power, they can reduce the reactive power by using these VSDs and VFDs. Normally in traditional case, they are controlling the heat air by using a gate, only a gate. But in case of factory 3, they are using these VSDs and VFDs to control the motor RPM of withering troughs instead of controlling the air. But in case of controlling air by a gate, we do not control the motor RPM, then we do not control the energy. However, the capacity of leaves, we are using the same motor. But in case of VSDs and VFDs, we can reduce the RPM levels according to the requirement, according to the capacity. So then we can reduce the energy or the KVA demand by a factory. So here is the saving of electrical energy in the factory 3. So according to that, their SEC, specific energy consumption, is less than the energy benchmark of low country as 0.74, and same manner their trough energy usage is less than factory 1. So therefore factory 3 has achieved the energy benchmark successfully. Conclusion, normally in daytime, 50 to 55% of the electricity is used, and calculated SEC for factory 1, 2, 3 are here, and less specific energy consumption was reported from the factory 3 under the energy saving techniques, and also apparent power also less in factory 3, and highest energy consumption was reported in the withering stage as the previous studies also. So we have to specially concern about the withering stage, and we have to put some kind of energy strategies to this withering stage. Finally, to smooth these techniques, energy management techniques, we have to do another studies by adapting to automated machineries and by achieving more sustainable techniques. Here are the references, and thank you.
Thank you, Ms. Pereira, for your informative presentation. Now, paper is open for the discussion. Any questions from the audience? You can even use the chat option if you have questions, or you can directly ask questions to Ms. Pereira. Okay, in the absence of questions from the audience, let me ask one or two questions from Ms. Pereira. So, in the presentation, you mentioned that even though Ceylon tea, which is the most finest tea in the world, but our cost of production is very high. But other, well-recognized tea-producing countries like India and China, they have low cost of production and low energy consumption and usage. So why? Can you explain what are the reasons behind this?
Yes, madam. Especially, they have used some kind of energy benchmarks for their productions, and they have established already some kind of energy benchmarks and precision practices that can be used for the production. Especially, they have identified what are the steps that are using highest energy for the production of black tea. At that manner, they have reduced the energy consumption and the cost of production in same manner.
Okay, thank you, Ms. Pereira. And one more question. Actually, low country is the highest producing tea producing region, but now in mid and high grown areas, the usage, energy usage and consumption is low. That's what I observed during the presentation. So what are the reasons behind this?
Yes, madam. In both, they have low production. Actually, the first reason, the production also low. So the energy consumption is then low. And also another thing is they are using more firewood for the production and more than the electricity, especially in traditional factories in upcountry. The factories are much old than 50 years in upcountry. They are traditionally using firewood for the production. So their electricity consumption is, as a percentage, it's less than the low country.
Okay, thank you. And one more question, actually, during your presentation, you mentioned that this energy
of black tea in precise manner that can be used our benchmarks also.
Okay, thank you, Ms. Pereira, for your informative presentation and answering the questions.
Thank you.
Okay, now we will move on to the second presentation. The title of the presentation is Analyzing Polymorphism of BG357 into H10 cross through bulk segregation process with selected DNA markers of rice blast resistant genes. The authors of the paper are W.A.D. Jayawardena, G.A.U. Jayasekara, K.L.N.S. Pereira, S.N.S. Mohotti. The paper is presented by W.A.D. Jayawardena. So over to you, Mr. Jayawardena. Sorry, sir. Sir, I can hear you. Yeah, over to you, Mr. Jayawardena, to present your paper.
Good morning to you all. I'm here to present my research work titled Analyzing Polymorphism of BG357H10 cross through bulk segregation process with selected DNA markers of rice blast resistant genes. Well, I would like to introduce myself. I'm Dr. Amma Jayawardena from Department of Plant Sciences, University of Colombo. Completed my PhD under the supervision of Professor Chayasekaran. To get started, let's see the importance of rice to the world. Rice is the main staple food in the world, and its production should be increased 50% by 2030 to meet the ever-increasing population growth. But this is quite challenging due to two reasons: climate changes and numerous rice diseases. Among those numerous rice diseases, rice blast is the most important rice disease that is caused due to a fungus called as macrophagocytia. Blast is normally controlled by using fungicides if the field occurrence is detected, which causes high production cost and environmental pollution. So the best way to mitigate the effect of blast is to identify blast-resistant cultivars. Conventionally, the blast resistance in the germplasm is identified through phenotypic screening. However, the phenotypic screening is influenced by environmental conditions, cumbersome and expensive. It also requires the use of lots of field spaces. Therefore, identification of blast-resistant genes with DNA markers tightly linked to them could save money as well as time that's spent on conventional methods. So the specific objective of the current research was to identify polymorphic DNA markers linked to different blast-resistant genes among selected resistant and susceptible parental population, F1 population, F2 population of BG257H10 cross. Let's move to the methodology. As I have already mentioned in the objective, the current research analyzed parental population, F1 population, F2 population of BG257H10 cross through bulk segregant analysis process in order to identify polymorphic DNA markers linked to different blast-resistant genes. So at the beginning of the research, we had to select parental pair to develop the cross. So based on the previous phenotypic screening data, we selected highly blast-resistant BG257 and highly blast-susceptible H10 as the parents for our cross. We used them to develop F1 and F2 population. These images shows how we perform those crosses. So the F2 seeds and the parental seeds were established in the well-designed plots at RRDI Batalagoda, Upland Blast Nursery. After F2 plants and the parental plants were grown to the proper stage, they were infected with blast pathogen by using spore suspension and infected dry leaves in order to identify the segregation pattern of F2 population. So this was basically done in order to identify the F2 population of ours harboring blast-resistant genes or not. If that's so, we could easily analyze this cross with selected DNA markers in the BSA process molecularly. So this was the specific design for that plot we used to establish the F2 plants in the infection. The infection process was carried out for 21 days of period. At the end of the 21 days of period, the F2 plants were scored for blast reaction by using IRRI stipulated blast scoring system. Moving to the methodology of the bulk segregant analysis, this is one of the newest technique that used in molecular biology. In this technique, it analyzes DNA bulks rather than the individual DNA. So we incorporated this technique for our research. In the bulk segregant analysis, at the beginning, we developed 5 DNA bulk by using the leaves of 5 population of BG257H10 cross. Those 5 DNA bulks were resistant parent DNA bulk, susceptible parent DNA bulk, F1 DNA bulk, F2 resistant plant DNA bulk, F2 susceptible plant DNA bulk. These aforementioned 5 DNA bulks were subjected to PCR by using 13 DNA markers. These 13 DNA markers were identified by a genealogy study that was carried out at RRDI Batalagoda prior to this research. Out of these 13 DNA markers, 12 were SSR markers, one was a gene-specific marker. Then the PCR product that obtained for all 5 DNA bulks of this cross with 13 DNA markers were firstly run in 2% agarose gel. Later, the same PCR product were run in 4% polyacrylamide gel in order to identify the allelic band sizes. However, the polyacrylamide gel electrophoresis was not performed for YL15587 marker as it was a gene-specific marker. So that's completes the entire methodology for the current research work. Let's turn to the result that we gained in our research work. Firstly, I will start off with the result that we gained for the filter screening of F2 population. In the segregation pattern analysis, it determined that the segregation pattern for the F2 population was 169 to 42, 169 blast resistant F2 plants and 42 blast resistant F2 plants. So based on the segregation pattern or the ratio, it indicated that the presence of more blast resistant F2 plants in the F2 line was most probably due to the presence of blast resistant genes that controlling the resistant phenotype in the F2 population. And these blast resistant genes were contributed to the F2 population from blast resistant parent plant BG357. So we were very confident about the result that we're going to get in the bulk segregant analysis process after this outcome. Let's turn to the result in the molecular analysis. In the bulk segregant analysis, as I have already stated, we analyzed BG357 H10 cross with selected SSR markers and one gene-specific marker. At the end of the bulk segregant analysis, out of 13 DNA markers, we found 2 polymorphic DNA markers, namely RM102 and YL15587, that clearly differentiated the 5 population of this cross: parental population, F1 population, F2 resistant and susceptible plants based on clear DNA polymorphism. Rest of the DNA markers were non-polymorphic due to various reasons. So these were the gel images that we obtained for those 2 polymorphic DNA markers. Moreover, YL15587 gene-specific marker reported to be located in the beta blast resistant gene. On the other hand, RM102 marker linked to PI20 blast resistant genes. So all in all, at the end of the current research, we identified 2 polymorphic DNA markers, namely YL15587 and RM102, that can be used for the direct selection of blast resistant genes, theta and PI20, when these genes are transferring from blast resistant parental rice variety BG357 to another in future rice breeding programs. So these were the references that we used in the current research. Before concluding, I must acknowledge my supervisor, Professor Anil Jayasekara, who helped me immensely throughout this research work, and rest of the institute who involved and collaborated in the research. Plant Science Department, University of Colombo, Rice Research and Development Institute, Batalagoda, GeneTech Molecular Diagnostic Laboratory, and HETC project for sponsoring our research. That completes my presentation. Yes, thank you all for your attention.
Thank you, Mr. Jayawardena, for your very interesting and very scientific, informative presentation. And now paper is open for the discussion. Any questions to Mr. Jayawardena? I prefer to have actual questions from the audience. Any questions to Mr. Jayawardena? Okay, in the absence of questions from the audience, let me ask one or two questions. So in the presentation, you mentioned that this is a novel technique you establish in the country, right? Mr.
Yes.
Yeah. So can you use the same technique to identify the blast-resistant traditional rice varieties in the country?
This technique can be used for not only for the blast, even for the other rice diseases as well, such as bacteria, blight, brown spot, so and so. Even for the traditional rice varieties in Sri Lanka, you can use this technique. But first of all, before using the technique, we have to see by using the parental rice varieties whether the markers are working, because we are taking markers from outside, from other countries, and taking here. And the markers were developed by using different rice varieties. So first of all, we have to see the markers are working. Then definitely we can.
It's very interesting. Actually, one more question. Actually, now in Sri Lankan conditions, in improved varieties, they know what are the blast-resistant rice varieties. So how they have, come to such conclusion, whether it's through phenotypic screening?
Yeah, till now, only a few research were conducted in blast, though it is the major rice disease in the world. One of the reason is in Sri Lanka, the main rice disease is not blast. The main concern is upon the bacteria blight. So they are not more towards the research on the blast. And the other reason is up to now, only few researches were conducted in blast, that also by Dr. Nimal Disanayaka, the former director of Rice Research Institute of Bathalagoda, and it was based on the phenotypic screening. It was not based on the DNA work. So what we have done in our research, we combine the DNA work with the phenotypic screening, so we can easily identify the superior genotype at a very earlier stage, so we can use them for breeding programs rather than waiting for years to get the superior genotypes. So DNA markers assist us. It reduces the cost of labor, it reduces the time, and we don't need large field spaces. Even the off-field seasons, we can use DNA markers and we can carry out our research and we can identify the superior genotypes.
Yeah, it's very interesting. Actually, how long normally for this analysis you take? How long in the lab for do this analysis? Can you make it to a form of a kit, something like that, which is very user friendly?
Yeah, the techniques are very, very improved nowadays. Even we are not doing the DNA extraction by using the traditional DNA protocols such as doily and doily methods. We use kits. So the DNA extraction takes place within 1 hour, so you can analyze if you have the plant materials on your hand. But in my case, what happened, I didn't use any DNA kits. I have to do it in my own, and it
can't you, from your research finding, can't you, make this into a form of a kit so that it's easy for the scientists to do screening, something like that? That's why I'm suggesting.
Now, the problem is now still Sri Lanka, we are not into that area that combining.
Yeah, you can collaborate with some, Okay,
First
thank
of
you.
all, we have to change the system and then we have to go for the rest of the things.
Yeah, thank you, Mr. Jayawardena. It's very interesting and very scientific presentation. Okay, we have 2 more presentations in the technical session number 1. And those 2 presentations will be chaired by Dr. Jayani Wevel. Over to you, Dr. Jayan.
Good morning to you all.
Thank you, Dr. Kanchana. So the next presentation will be on fatty acid profiles of selected non-pigment
New Improved Rice Varieties, Oryza sativa of Sri Lanka, authored by MD.W. Samarasekara, WKSM Abesekara, KRR Mahanama, IGN Heva Judike, HPPS Somasiri, DMJ B Sena Naik. And the presenter is MD.W. Samarasekara. Over to you.
I am Madara Samaranayaka, and the title of my presentation is Fatty Acid Profiles of Selected Non-Pigmented New-Improved Rice Varieties of Sri Lanka. These are the other authors, and this study was funded by Sri Lanka Treasury. This is the content of my presentation. Let's move on to the introduction. Rice is a popular cereal crop growing across the world, and it is the dietary staple for nearly half of the world population. Rice reported to provide significant quantities of carbohydrates, proteins, and also fat for millions of people living in Asian countries. Rice fat consists of high quantities of monounsaturated fatty acids and polyunsaturated fatty acids compared to saturated fatty acids. In Sri Lanka also, rice is the dietary staple, and per capita consumption is around 114 kilograms per year. There are thousands of traditional and more than 60 new improved rice varieties available in the country, and widely cultivating and consuming varieties are the non-pigmented new improved rice varieties. And nearly 70% of the rice cultivation in Sri Lanka consists of non-pigmented or white rice varieties. However, investigations on fatty acid compositions of rice are extremely limited. Therefore, the objective of the present study was to evaluate fatty acid profiles of some popular non-pigmented new improved rice varieties widely cultivated in Sri Lanka. Let's move on to methodology. As for the materials, locally grown 4 non-pigmented new improved rice varieties, namely BG450, BG366, BG379-2 and BG94-1 were collected from Rice Research and Development Institute, Batalagodi, Sri Lanka. Actually, this is a part of a research project, and in the whole project we had studied fatty acid profiles of 25 rice varieties. Except the fatty acid profiles of these 4 varieties, others are already published. Therefore, today in this presentation, I am presenting only the fatty acid profiles of 4 rice varieties which are not published elsewhere. Determination of grain sizes. Grain sizes of rice varieties were determined according to the internationally accepted methods and classified as extra long, long, medium and short grains based on the following classification. Determination of fatty acid profiles. For the determination of fatty acid profiles, rough rice samples were dehulled and ground to pass through 0.5 mm sieve. Then fat was extracted from rice flour using automated Soxhlet extraction method. Then extracted fat was derivatized to fatty acid methyl esters by rapid transmethylation method using methanolic KOH, and derivatized fatty acid methyl esters were analyzed by gas chromatography flame ionization detection method. Let's move on to results and discussion. This table shows the grain length and sizes of studied rice varieties. According to the grain size classification used, there were short, medium and long grains. This table shows the monounsaturated fatty acid profiles of studied rice varieties. According to the results, total monounsaturated fatty acid content of studied rice varieties varied from 9.95 to 13.60 milligram per gram of rice, and there were 3 monounsaturated fatty acids, and most abundant monounsaturated fatty acid was oleic acid, and its content varied from 9.57 to 13.12 milligram per gram of rice, and it was highest in short grain variety BG450. This table shows the polyunsaturated fatty acid profiles of studied rice varieties. The total polyunsaturated fatty acid contents of studied rice varieties range from 8.32 to 11.26 milligram per gram of rice, and there were 4 polyunsaturated fatty acids, and most abundant polyunsaturated fatty acid was linoleic acid, which is considered as an essential fatty acid, and the linoleic acid content varied from 8.05 to 10.95 milligram per gram of rice, and it was also highest in BG450 rice variety. These are the saturated fatty acid profiles of studied rice varieties. According to results, the total saturated fatty acid contents varied from 4.63 to 7.04 milligram per gram of rice, and there were 4 saturated fatty acids, and most predominant saturated fatty acid was palmitic acid, and it ranged from 4.13 to 6.24 milligram per gram of rice, and it was lowest in long grain rice variety BG941. Overall, the studied rice varieties had more monounsaturated and polyunsaturated fatty acids rather than saturated fatty acids, and monounsaturated fatty acids were the highest. The total unsaturated fatty acid contents of studied rice varieties varied from 18.27 to 24.86 milligram per gram of rice, and total unsaturated fatty acid content was also highest in BG450 short grain variety. In conclusion, the fatty acid profiles of studied rice varieties varied significantly among the rice varieties, and the highest monounsaturated fatty acid, polyunsaturated fatty acid, and essential fatty acid linoleic contents were exhibited by short grain variety BG450. The rice varieties with healthy and nutritionally important fatty acid profiles could be used as raw materials for developing functional food products, and also may use as genetic sources for breeding new varieties with high nutritional qualities, and the data generated may contribute to the development of National Food Composition Database of Sri Lanka. These are the references used in the present study. And finally, I would like to thank Sri Lanka Treasury for its financial support, and Dr. Sena Nayaka, the Director of Rice Research and Development Institute, Battalagoda, Sri Lanka, for providing rice samples for the study. Thank you.
Okay, thank you, Ms. Samrasekara. The presentation is open for questions. Hello? Hello? Can you hear me? Hello? Can you hear me? Okay, so the presentation is open for questions. Any questions from the audience? For Ms. Samranayaka? No? Okay. So I have one question. So you said like BG450. So the short grain variety has main impact for the saturated and unsaturated fatty acid. So is there any other physical parameters of that grain that would go for the content of the saturated and unsaturated fatty acid rather than the size of the grain? Have you any idea? No.
I haven't checked the other parameters, only the grain size and shape.
Okay, so need more research on that, right? Yes.
Yeah.
Okay. Okay, thank you. And then, so what do you think? Like, so there is actually a difference between the saturated fatty acid and the unsaturated fatty acid, right?
Yes.
So, but in BG450, as in the presentation, so both has the similar effect in this variety, BG450. So, do you think what are the health benefits of the saturated fatty acid versus the unsaturated fatty acid?
Actually, the unsaturated fatty acids are beneficial. Mostly, they contribute to protection against coronary heart diseases, but saturated fatty acids, they contribute to coronary heart diseases. It's better if you have more unsaturated fatty acids and less saturated fatty acids in rice varieties.
Okay. But in BG450, according to your results, so that is that has both saturated and unsaturated fatty acid in higher content. Is that right? Yeah.
Yes. Here I have only mentioned the monounsaturated, saturated, and polyunsaturated fatty acid contents. And there are some nutritional indices also which we can use to evaluate the fatty acid profiles of nutritional impact of these varieties, fatty acids. Some are thrombogenic index, then ratio between hypocholesteremic and hypercholesteremic fatty acids. I haven't mentioned those values in this presentation, and the polyunsaturated to saturated ratio. You can take those values to evaluate the impact of these rice varieties. If you according to those indices, actually the BG 94-1 variety was better. Yeah.
Okay. Okay. Thank you, Mr. Samranayaka. Thank you for the presentation.
Thank you.
So the next. Okay. So the final presentation for the technical session 1 is on like isolation and identification of native antagonistic yeast as biocontrol agents of post-harvest fungal pathogens, authored by E.A.M. Jayasinghe, U.M.A. Kumara, K.D.N. Rodrigo, and N. Tirukacheva. So the presenting author is KMDN Rodrigo. Over to you, Rodrigo.
I welcome you all to my research presentation, and I am Dilsha Rodrigo from Department of Agricultural Technology, Faculty of Technology, University of Colombo. The research presentation is on the title Isolation and Identification of Native Antagonistic Yeast as Biocontrol Agents of post-harvest fungal pathogens. Now let me take you through the background of this research. Sri Lanka, as a developing country, has been experiencing many economic losses due to high amount of post-harvest losses which take place from the stage of the field until they are dispatched to the market. Fruits and vegetables, being perishables, are highly susceptible to these post-harvest losses owing to many reasons. And among all these reasons, the occurrence of post-harvest diseases has become a leading cause for a considerable loss of these fruits and vegetables. Also, the fungal diseases cause major crop losses ranging around 50%, especially in developing countries. Therefore, as a remedy, farmers have been using synthetic fungicides for a longer period of time, and now it has brought out many adverse effects both on the human health as well as on the environment. Therefore, now it is a timely need to discover antagonistic use from the natural sources because it is very important to us as an eco-friendly and also non-fungicide method. Now let me take you through the objectives of this research. Main objective was to isolate and identify native antagonistic yeast from the natural sources against these post-harvest fungal pathogens in perishables. Whereas the specific objective was aligned to determine their in vitro efficacy of the antagonistic yeast from these natural sources. Now, let me take you through the methodology which was followed when performing this research. At the first, the collection of plant and soil samples from the natural sources. Here, the flowered plants and also straw, soil samples, leaves, plant exudates and plant sap were used as the samples and they were collected from an organic farm in Anuradhapur area. Also, after collecting these plant and soil samples, they were cultured in yeast extract peptone dextrose media, which was used to culture them, and also by following the subculturing and incubation until obtaining a pure colony of the yeast. Also, after that, the dual culture assay was performed, and after that, we were able to identify the killer yeast followed by the incubation. The mean growth rate of the fungal mycelium was calculated in order to observe the antagonistic ability of these potential antagonistic yeast. And thereafter, the antagonistic yeast isolates were identified. Further, 5 selected antagonistic yeast were chosen and also the DNA of them was extracted. After that, gel electrophoresis, PCR amplification was performed using D1 to D2 domain regions of 26S rDNA for further molecular identification. Also after that, the PCR products were sequenced and also they were analyzed by BLAST software for the further confirmation of these isolates. So, at the end of this research, the research findings could be outlined like this. Basically, 22 isolates could be isolated from the natural plant and soil samples, while majority of them were from the plant-derived substances. And also, here their color, appearance, growth rate, growth pattern, and also their colony morphology were significantly different from one to another. Also, among the isolated killer yeast, there were 5 killer yeast isolates which were selected as KY1, 4, 11, 15 and 20, which were found to be stable even in the in vitro conditions. So this is one of the best figures which were captured, which clearly shows the inhibitory zones which were developed by 3 different natural killer yeast isolates, namely KY15, KY4 and KY20 against Polytrichum gliosporidae, especially which is the causal pathogen in papaya and anthracnose, and also Aspergillus niger, especially found in black onion mold, and also the Polytrichum gliosporidae, especially found in papaya stem entrail. Also, this figure clearly depicts the clear inhibition zones which against the fungal pathogen. So it is very vivid that the selected isolates were good enough to show their antagonistic activity against these fungal pathogens. This gel image, which was provided by PCR applicants of D1, D2 regions from 26 rDNA of these 5 different killer yeast isolates, were also supporting the finding that these isolates were also stained, and thereafter the particular isolates were clearly visible and their applicants were found. Also, according to these DNA homology results, it is very clear that they align with the antagonism of KY isolate 1, 4 and 11 against the causal pathogen Aspergillus niger, reflecting them as similar homology with PGR, Kudryas sevi and also PGR species. Also, thereafter, the antagonism of KY15 and KY20 was also proven against the Polytrichum gliosporidae, which showed homology with Trichosporum acai and also Trichosporum infestans. So, finally, the research conclusions can be set under the ability to use them, especially T. acai and T. infestans, as antagonists against anthracnose as well as stem endraft in papaya, while PGR Kudryas baby has a potential against onion black mold pathogen. Therefore, at the very end, it can be concluded that these are the potential antagonists which can be highly used in upcoming research sessions as well as for the betterment of this country. So, especially the fungal pathogens, mitigation is viable with these potential antagonists, and also these are the references which were used to grab the background knowledge, and also this is the end of my presentation. Thank you very much for your kind cooperation. Thank you.
Okay, thank you very much, Radhika. So the presentation is open for discussion. Any questions from the audience? Okay, in the absence of the questions from the audience, so I will ask one or two questions. So So have you checked the content of the sample when you're collecting it from the Anuradhapur area?
Yes, madam. The thing is that when we were collecting the natural samples, what we did was we just searched for the raw samples, right? Like straw, right? And also the phloem sap, and also the leaf washing water, right? So we were not sterilizing the plants, right? And the point of using an organic farm, it was to assure that these fungus are not get killed. That's why we went for choosing an organic farm. In the initial stage, we didn't use any sort of preliminary application for that.
Okay. So as you mentioned, like the majority of the substance on the collection was plant, right?
Yes, madam.
Okay.
Most of them were from inside the plants,
Plant.
stuff like that.
Okay. So, right. Okay. Then, so what was the when you isolating these fungi from the samples that you collected? So, how many days that you needed to incubate, and what were the temperatures?
Yes, we incubated them for about 4 days at 28 centigrades. That were the conditions that we applied for when isolating these fungals.
Okay. So after isolating, when you apply it for to find the antagonistic activity of the different pathogens,
Yes.
how did you do that?
We used the method dual culture assay. So first we plated the fungal pathogen in the middle of the plate. So then by taking the isolates, first before going that, we made a grid and confirmed these pure cultures of these isolates. And what we did was we just placed them on the Petri plates around 1 centimeter away from the Petri plate wall. That is the dual culture assay was performed in order to find whether there is an antagonistic effect against this fungal pathogen.
Okay, thank you, Ratuko. And that was the end of the technical session 1. And I want to thank all the presenters who was in the technical session 1. And we can move to the next session. Okay, thank you, everyone.
Thank you very much, Dr. Veeralwala, and all the presenters. Thank you very much. I would like to thank Dr. Veeralwala and Dr. Abhesekara for sharing that. That marks the end of the technical session 1. Now, we are about to start the technical session 2. In order to chair the technical session 2, I would like to call Professor Chamini Hemachandra and Dr. Purna Piyatilaka from the Department of Environmental Technology.
Good afternoon, everyone. So welcome you all for the technical session 2 of the Annual Research Symposium of Faculty of Technology, University of Colombo. So before starting the technical session 2, I would give some overall introduction about the session. So altogether there will be 6 presenters in this session, and 3 presentations will be chaired by me, and the rest of the 3 presentations will be chaired by Professor Shamini Hemachandra. And all the presenters will have 10 minutes of time for the presentation and 5 minutes for the question and answering session. And yes, after each presentation, all the presenters will have undergone for the question and answering session. So I would like to introduce the first presenter of the session. The first presenter is presenting under the topic of isolation and characterization of thermostable alpha amylase enzyme producing bacteria from Mahaoya Hot Spring, Sri Lanka. The authors are H.D.D. Sadeepa, P.M. Manage, and K.A. Sirisena, and the presentation will be conducted by Ms. H.D.D. Sadeepa. Over to you, Sadeepa, and wish you good luck.
Hello everyone, I am Dilli Sadeepa from University of Sri Jayawardhanapura. Today I am here to present you my research regarding isolation and characterization of thermostable alpha amylase enzyme producing bacteria from Mahaoya Hot Spring, Sri Lanka. This is the lineup of my presentation. Let's move to the introduction. In industrial settings, they convert their substrate into more valuable end product using a sequential process which include physical, chemical, and biological transformation steps. There, in order to maximize the industrial output, they use harsh conditions in industrial settings, such as high substrate concentration, extreme pH and temperatures, non-aqueous conditions, etc. As well as they use chemical catalysts in industrial settings for maximizing industrial output. But the excess usage of chemical catalysts causes chemical waste, which cause several environmental problems such as air pollution, water pollution, soil pollution, etc. Therefore, it's essential to minimize the chemical waste while maximizing the industrial output. For that, microbial utilization has been identified as a better solution towards green chemistry. But if we use microbes or microbial products in industrial setting, these microbes or microbial products should be able to overcome harsh conditions used in industrial settings. For that, extremophilic microorganisms which produce extremozymes have been identified as a better solution. As an example, if we can use thermophilic microorganisms which produce thermostable enzymes in industrial process which operates under high temperatures. Microbes have huge gene diversity and they keep genes code for carbon degradation enzymes, starch degradation enzymes, cellulose degradation enzymes, hemicellulose degradation enzymes, chitin degradation enzymes, pectin degradation enzymes, and other carbohydrate degradation enzymes, protein degradation enzymes, etc. Therefore, if you could identify microorganisms which produce industrial important enzymes which can tolerate harsh conditions used in industrial settings, we can use those microorganisms in industrial settings successfully. Therefore, the current study focused on isolation and identification of thermostable alpha amylase producing bacteria from Mahaoya Hot Spring. Let's move to the materials and methods. Water samples were collected from Mahaoya Hot Spring. During the sampling, water samples were collected from both surface and bottom of the hot spring. Physical parameters of the water samples such as pH, temperature, dissolved oxygen levels were measured at site itself, and samples were transported to the laboratory in ice boxes as soon as possible. Then the samples were enriched in quarter diluted nutrient broth supplemented with soluble starch, and bacteria were isolated. The isolated bacteria were screened for the alpha amylase production, and crude extracts were screened for the pH stability and temperature stability. The initial screening was done using spot test on starch agar, and secondary screening was done by using DNS method. Enzyme activity was expressed as the amount of enzymes that release 1 microgram of glucose from the starch per minute. Then the temperature stability, pH stability, and optimum incubation time for the microbial growth was examined. Then the most potential bacteria were identified using 16S rRNA gene sequencing method. There, microbial DNA was extracted using heat-cool method, and PCR amplification was done using universal primers for bacteria. Then the PCR product was run on an agarose gel referring to a 100 pp ladder. 16S rRNA sequencing was done at Macrogen, Korea, and resulted sequences were trimmed and removed primer sequences using bioedit tool. Then the bacterial isolates were identified using manual blast tool in the NCBI. Let's move on to the results and discussion. These are the water quality parameters of the hot spring. The temperature of the hot spring was recorded in a range of 53.7 degrees of Celsius to 55.4 degrees of Celsius. Slightly alkaline pH values were recorded, and during the initial screening for the alpha amylase activity, 4 bacterial isolates were positive for the enzyme production. Among those bacteria, MH1 and MH27 exhibit the most promising results, and those 2 isolates were used for the further studies. This figure shows the glucose standard curve used for enzyme activity measurements, and it's a linear graph with 0.99 R-square value. This graph shows the effect of temperature on enzyme activity of fruit enzyme extract of MH1 and MH27. The optimum temperature for MH1 was 45 degrees of Celsius, and for MH27 it was room temperature. But the highest productivity was observed from MH27 at room temperature. This graph shows the effect of pH on enzyme activity of fruit enzyme extract of MH1 at 40 degrees of Celsius and MH27 at room temperature. The optimum pH for MH11 and MH27 was 4 and 7 respectively at their optimum temperatures. This graph shows the optimization of incubation time for microbial growth. There, the optimum incubation time for both MH11 and MH27 bacterial isolates were 36 hours. This is the PCR cycle used for PCR amplification of genomic DNA of bacterial isolate. This is a photograph of agarose gel electrophoresis of PCR products. The bacterial isolates MH11 and MH27, which produce alpha amylase, which stable at 45 degrees of Celsius and 30 degrees of Celsius respectively, were identified as Bacillus cereus strain BBE0 and Bacillus thuringiensis strain ISJ33. Finally, the results were concluded that hot spring are a rich source of thermophilic bacteria which produce alpha amylase enzymes, and bacterial isolates MH1 and MH27 isolated from Mahawe hot spring could be useful source of alpha amylase enzyme which can be successfully used in industrial settings. These are some of my references. Finally, I would like to acknowledge University of Sri Jayawardhanapura for the financial support and Center for Water Quality and Algae Research. Thank you very much.
Thank you, Ms. Sadeepa, for your excellent presentation. And now the presentation is open for discussion. Okay, until some others are preparing for the questioning, I am having some questions. Ms. Sadeepa, can you explain whether the primers you used for the study, were they designed by yourself or were they available in the published literature?
Actually, madam, they were available in the published literature. I used 16S rRNA gene sequencing for identification. For that, I used universal primers for the bacteria, but I sent DNA samples instead of PCR product for the sequencing. They used the same primer for the PCR amplification, according to my request.
Okay, so sometimes I maybe missed that part. Can you define the amplicon size of the PCR?
1,500 people.
Okay.
Is the product.
Okay.
The V3 and V4 region of 16S rRNA gene.
Okay. So, are there any other questions?
Ms. Sadeepa, I am Dr. Abhesekara here. Just a clarification, the enzymes that you extracted, is that thermostable? Because some alpha amylases you can even use at 100 degrees. Any idea about it?
Yeah, I extracted those bacterial enzymes from Mahawe hot spring. The temperature of the hot spring was about 55, around 55. And I tried to isolate thermostable amylase, cellulase, and several other enzymes in my studies. Here I'm just focused on amylase enzyme. It's only stable at the optimum temperature was 45 degrees of Celsius. But I could use those enzymes even at 60 degrees of Celsius. They were stable, but the optimum activity was observed at 45 degrees of Celsius.
Okay, one more question. As a positive control, did you use commercially available alpha amylase?
Yeah, so I used commercially available alpha amylase as positive.
So that should be, I think, in your coming publications. Please include that one also, because the activity has to be compared to the positive control in the experiment.
Yes, madam. Thank you.
Okay. Okay. Thank you very much.
Thank you, Dr. Abhesekara. Are there any other questions from the audience? Okay, with the absence of any other questions, we can proceed for the next presentation. Thank you, Ms. Sadeepa. The next presentation, sorry, are there any questions? Okay, next presentation will be on studying the effect of spatio-temporal land use, land cover, LULC, dynamics on inundation occurrence in Bolgoda River Basin, Sri Lanka. The authors are BLN Nadishani, VPNA Premasiri, TANT Pereira, RPWN Arunasiri et al. And the presentation will be conducted by Ms. VPNA Premasiri. So, Ms. Premasiri, over to you.
Floods affect socio-economic and environmental well-being of the society. Floods are inevitable but can be mitigated by managing its causes and consequent impacts. With that note, let me start my presentation. I'm Lilu Pavignana from the Department of Environmental Technology. Our study is titled on Study the Effect of Spatio-Temporal Land Use, Land Cover Dynamics on Inundation Occurrence in Volgodi River Basin, Sri Lanka. Moving into the introduction. Floods are the most frequent natural hazard among all other natural disasters in Sri Lanka. In recent past, there's a significant increment in flash floods, which critically emphasizes the timely requirement of studying floods, its occurrences, consequent impacts, and its causes. Land use and land cover changes have been identified as one of the most considerable causes to the increment of flood risk in Sri Lanka. Volgodi River Basin has been identified as more flood-prone area, especially for flash floods. These flash floods mostly during southwest monsoon and second inter-monsoon rains. According to the Japan International Cooperation Agency, land use change has been identified as one of the main causes of inundation in Volgodi River Basin. In this table, you can see other main causes of inundation in Volgodi River Basin. So, why this study is important? There are very few studies have been undergone on Volgodi River Basin. Most importantly, no any systematic study on the impacts of LULC changes on flooding in Volgodi River Basin. Thus, a detailed study is essential on LULC changes and inundation fluctuation in Volgodi River Basin. So, the specific objectives of the present study was therefore to quantify LULC changes during past 30 years in Volgodi River Basin and investigate the relationship between LULC changes and flood occurrence and hazardous level. Moving into the methodology, Volgodi River Basin is located in southwest of Sri Lanka, which spreads over both Kalutara and Colombo districts of Western Province. Volgodi Basin comprises of two major water bodies, which are Volgodi South Lake and Volgodi North Lake. When considering the land cover in Volgodi River Basin, both lakes are surrounded by home garden, urban areas, scrublands, crop cultivation, and mangroves and so on. In the literature, we can find out most of the agricultural lands and grasslands near the Volgodi Basin have been converted into built-up areas due to urbanization. Here you can see in this map our study area. In the study, two types of data were obtained. Primary data were collected through field observation, discussion, workshops, public meetings, and questionnaire survey. Firstly, 9 flood-affected DS divisions were selected after discussing with disaster management and disaster control units of DS divisions of Volgodi River Basin, and we have conducted 3 workshops to collect information on inundating areas, roads, historical fluctuation of inundation, causes of floodings, and as well as the environmental issues of floods. Furthermore, a comprehensive questionnaire was prepared as an electronic form to gather data on inundation fluctuation. Inundation fluctuations are determined by using 4 parameters. The parameters were frequency of floods, duration of floods, area of flooding area, and the depth of floods. As well, to this comprehensive questionnaire survey, we collected the information about community perception on flooding in Walguru River Basin. Secondary data were collected through satellite images and land cover data collected from USGS Earth Explorer official website and Google Earth software. Here you can see a flow diagram of secondary data collection and generation. The first step was satellite image acquisition. We used 4 satellite images in 1989, 1997, 2008, and 2019. The second step was NDVI calculation and map extraction. For that, we choose Landsat 5 and 8 images. The ArcMap software we use for that. The third step was NDVI map classification. The classification done under 6 categories. The categories were forest and scrublands, agricultural land, grasslands, built-up areas, abandoned areas, and water bodies. NDVI classification was done using ArcGIS software. Finally, area and change detection metrics were calculated. This all data were analyzed using Excel and SPSS statistical software. Moving on to the most important part of the presentation, results and discussion. Here you can see classified NDVI maps in 1989, 1997, 2008, and 2019. Here you can see the all 6 categories were determined in this study and how these categories, these land covers were changed over the years, over the 30 years of study time period. Here you can see the detail, the calculated data from the NDVI maps. Here you can see 6 categories of 6 categories, and how these categories, the area percentage have been changed over the years. Here we identified that vegetation cover has been decreased by 4,345 hectares, whereas the build-up area has been increased by 5,667 hectares during the study period. And you can see how the area percentage as well as the area in change in area in hectares. Moreover, discussing the area, land cover and land use change during the period of 1989 to 2019, build-up areas were increased from 1,749 hectares to 7,429 hectares. That means 80% from total area. Total vegetation cover, including forest, agricultural lands, and grasslands, were decreased in 12% from total area. Abandoned lands were decreased in 63% in total area, and water bodies were decreased in 17% in total area. it can be concluded that there's a distinct change in land use change, especially vegetation cover and build-up areas during the study period. Next, moving on to the inundation analysis. Here you can see how the inundation analysis parameters changed during the study period. Here, frequency has been highly changed during the study period, and duration of floods has been highly changed. Area of the floods has been highly changed, where the depth of floods has not been changed over the study period. Moreover, discussing the inundation analysis, most of the people responded that inundation frequency is now higher than past. As well, many people responded that inundation duration is also comparatively high now than past. As well, inundation area is now high comparing with the past. Comparing the past. So inundation frequency, duration and area have increased gradually, which indicating a strong positive correlation with built-up areas. Contrastingly, 42% people will say that inundation depth has not changed through the history line. Actually, inundation depth, which was less than 2 feet, has no gradual increase during the study period. Water stagnation level was high around Bolkoda South Lake area, actually, which is 2 to 3 feet. In here, we can conclude that inundation was highly caused by the accumulation of rainwater in the area than the stream overflows because of the disturbances caused by as a result of build-up areas due to the urbanization. Finally, these all findings highlight the critical necessity of sustainable land use planning and management to mitigate the flood risk in Bolkoda River Basin. With that, let me end my presentation. I acknowledge Department of Agricultural Engineering, Faculty of Agriculture, University of Ruhuna, Department of Environmental Technology, Faculty of Technology, University of Colombo, and all others who supported in numerous ways. Thank you very much for listening. Now you have time. Feel free to ask any question regarding the presentation and our study.
Thank you, Ms. Premasiri. Now the presentation is open for discussion. So I'm having a question. So Ms. Premasiri, can you just explain us, because now what I understand is you are having prevailing data for some years back, and can this data project the future incidents, I mean future levels of floods in this area, future inundation capacities of this area, can it be done with your data?
Yes, madam. The future flood occurrences can be predicted through flood forecasting using flood forecasting softwares.
I mean, from your data, you can do that at least.
Yes, madam. Yes, madam.
That's good. So you suggested some sustainable land use patterns and planning of the management of the area, such things. Can you just explain for the audience that what are the possible mitigation methods to manage that area according to your knowledge? Can you just suggest?
When vegetation cover, we can cultivate suitable crops, and when it comes to the build-up areas, when we are building and do new constructions, we can think about the water flowing and like that, but like drains, and we can build up drain systems, better functioning drain systems, like that kind of things, through that kind of sustainable buildings and construction projects, we can manage the land.
Yes, other than that, actually, can I answer to that question?
Sure, sir.
Okay, so I think there's a big, huge issue is the land use pattern changes. So you can see the last few years, the land use pattern changes. Urbanization is very high in this area. So one of the most important thing actually, so now we have to demarcate the area and restrict more the illegal land use changes actually. So this is one of the most important part, because this is a highly urbanized area and highly populated area. So therefore we have to manage with the illegal land management. So this is one of the most important thing. So we have to do it in this area.
I think that is the most important part of this study. Explaining the importance of the study. Thank you, sir, and thank you, Ms. Premasiri. Are there any other questions from the audience? Okay, with the absence of any other questions, we can thank Ms. Premasiri for her excellent presentation. So I would like to move to the third presenter of the session. The topic is assessing the human perception on urban home gardening as a sustainable approach. The authors are D.P. Lakmini, T.A.N.T. Pereira, R.P.W. N. Karunasiri, V.P.N.A. Premasiri, H.M.M.S.D. Herat, and R.U.K. Piyadasa. The presenter is D.P. Lakmini. Over to you, Lakmini, and good luck for your presentation.
Hello everyone, I am Ms. D.P. Lakmini, presenting author from Department of Environmental Technology, Faculty of Technology, University of Colombo. Today I am presenting the research findings entitled on Assessing the Human Perception on Urban Home Gardening as a Sustainable Approach. Let's move on to the introduction part. As you all know, food insecurity is a rapidly growing issue worldwide. It is the disruption of food intake due to lack of access to enough good, healthy and quality food. It means you have food but not in adequate amount, sufficient for a person. It may be due to lack of money or any other resources such as unavailability of quality food. Here you can see the difference between global primary crop production in 2000 and 2018. It was around 9.2 billion tons in 2018, which is 50% higher than in 2000. But more than 148 million people suffer from hunger in 2020 over 2019. Why? Because of the rapid increase of world population. So, this primary crop production is not sufficient to fulfill the requirement. Asia represents the 2.37 billion global population facing moderate or severe food insecurity, while over 1/4 of the population in Sri Lanka lack of sufficient and nutritious food to maintain a healthy lifestyle. Obviously, you know, agricultural sector of Sri Lanka mainly contribute to national food security. But these factors, availability of agricultural land, water stress, erratic weather and other factors detrimentally affects the food production. Especially urban sector of Sri Lanka remains food insecure and severely suffer from nutritional challenges. Therefore, to ensure food security, sustainable home gardening should be promoted to develop a
sustainable agricultural system. To overcome this urban food insecurity, we need to promote urban agriculture with novel technologies. Because in urban areas, we have very limited resources such as availability of space, soil, and etc. So we need to fully utilize these limited resources with the help of these technologies. Aeroponics, hydroponics, automated systems, balcony farming, and technological innovations such as apps, softwares, and sensors. Okay, what are the merits of sustainable home gardening? It may advance food security while strengthening the urban agri-ecosystem. Meanwhile, it improves land utilization, household socio-economic factors, and human well-being. Therefore, this survey research was carried out with the following objectives: to identify the existing urban agricultural practices in Colombo district, Sri Lanka, and to assess benefits and challenges for urban farming according to the perspective of urban home gardeners. Okay, then we'll see the methodology. We selected Colombo district as our sampling area. Total sample size was 402 home gardeners who attached to the ongoing urban agriculture project of Sri Lanka, Saubhagya 2020-21. And they were selected for the survey through random sampling from the 9 agrarian development centers. This will be shown in next slide. Okay, then the survey was conducted using mobile ODK Collect app to determine what extent the urban sector engage in sustainable agriculture and their perception on urban farming. Finally, the collected data were analyzed using Minitab. As I mentioned earlier, here the table shows the number of individuals surveyed from each agrarian development centers, namely Kotte, Maharagama, Homagama, Kahatuduwa, Kasbawa, Kolonnawa, Padukka, Kosgama, and Malabe. And this map shows the distribution of each home gardeners through the surveyed area. Now the results and discussion part. As you can see, this pie chart indicated that majority engaged only in conventional farming, while around only 9% focus on farming with novel technologies. So, the existing urban agricultural method is conventional farming. This bar chart represents the irrigation method used in urban farming. Majority of the urban home gardeners used pipe water to irrigate their crops, while only 4.48% farmers used rainwater as the main irrigation method. As figure 4 shows, organic fertilizer usage in Colombo district were 20%, 20.15%, while integrated fertilizer, it means both chemical and organic fertilizers, were used by 74.38% for the home gardeners. As highlighted in figure 5, majority of urban home gardeners, 51%, engage in home gardening with the purpose of home consumption. The least of respondents do home gardening for selling their yield. Further, 38 individuals expect to consume and surplus amount to sell. According to the perspective of urban home gardeners, main challenges for home gardening were high cost of inputs and less knowledge on gardening. Here, lack of suitable conditions is not a main issue for all, because majority disagree to this. As this chart shows, reduction of household food expenditure, extra monthly income, enhancing the value of land, recycling the waste, and reusing the wastewater
are the main economical and environmental benefits from home gardening according to the respondents. Finally, this shows majority of respondents, 89.55%, demand on organic food production in future, with considering economic and environmental benefits. Hence, it is a positive remark to introduce sustainable home gardening technologies to urban households. Last part, conclusions. So we can conclude this presentation like this. The study examined the potential of introducing sustainable urban farming to the urban sector of Sri Lanka to overcome urban food insecurity. The results revealed that although the existing farming type is conventional, majority of respondents like to contribute positively to the sustainable urban farming. As a strategy to overcome this urban food insecurity, urban domestic food production should be strengthened through enhancing sustainable home gardening using novel technology. Our earlier mentioned facts were based on these references. So it is the end of the presentation. Have a nice day. Thank you.
Thank you, Ms. Lakmini, for the excellent presentation. Now the presentation is open for discussion. Ms. Lakmini, can you explain, because I'm asking this question according to my personal experience. During your study, did you observe any trend of unregulated excess use of chemical fertilizers in these urban home gardening practices?
Actually, yes, madam. Mainly, their main fertilizer method was chemical synthetic fertilizers than organic fertilizers.
Yes, because most of the people doesn't know, as it is in a very little quantity, the growing area and the plant vegetations are in very little quantity. There may be a trend of having excess use of chemical fertilizers, and that can create another huge environmental problem. So, are there any educational programs under your study or in other institutions are carrying out any educational programs to educate these home gardening people around Colombo, in urban home gardening practitioners, according to your knowledge?
Actually, madam, currently, I think there's the educational programs, normally the awareness programs are going on, but
yeah, maybe there are no any regulations for that, but as graduates, fresh graduates, and people who are doing the research on that, I would like to suggest such awareness programs from your side, at least.
Yes, madam. Actually, we hope to, this is an initial study. So after analyzing these data, our next stage is to promote this urban home gardening. So in the future, we also hope to do these programs.
That's great. So are there any other questions?
Madam, I'm Sonali Herath here, one of the co-authors of the paper. Yeah. I also would like to add a comment to Ms. Lakmini's response for the question regarding the awareness programs.
Yeah, you are most welcome.
Yeah, actually during the study, we found that some of the awareness programs are conducted in the school level. Mm-hmm. So due to these programs, some of the school children are kind of interested in urban home gardening. That was a positive trend. But regarding the elder people, because this is the Colombo district and they all engage in the home gardening with their busy schedules, they are kind of reluctant to participate in the programs which are conducted in the agrarian development centers, mm-hmm. Which are mostly related with the paddy cultivators and those sector in the society. So regarding the other sector of the society who are engaged in the urban home gardening, there should be a particular awareness and new novel technologies introduced. That is a kind of future research and also a must thing to do in the future.
Yes, very importantly. Thank you, Ms. Heyrat, for adding that. Are there any other questions or any comments? Okay. So I would like to thank Ms. Lakmini again for her presentation and the answers. So after completion of the first three presenters, I would like to hand over the session to Professor Chamini Hemachandra to continue. Thank you, everyone. Okay, thank you very much, Dr. Purna Piyatilaka. So, good afternoon, you all. So, we will move on to the next presentation. The next presentation is on a remote sensing approach for assessing agricultural land use, land change using normalized difference vegetation index in Western Coastal Region, Sri Lanka. So, the paper is authored by J.A.C.J. Tilakaratna, R.P.W.N. Karunasiri, D.P. Lakmini, T.A.N.T. Pereira, V.P.N.A. Premasiri, H.M.N.S.D. Herath, R.U.K. Piyadasa, and G.Y. Jayasinghe. So the paper is presented by R.P.W.N. Karunasiri. Ms. Karunasiri, it's over to you.
I'm Marika Karnasari from the Department of Environmental Technology, Faculty of Technology, University of Colombo. I will be delivering this presentation on the research entitled A Remote Sensing Approach for Assessing Agricultural Land Use, Land Change using Normalized Difference Vegetation Index in Western Coastal Region, Sri Lanka. This presentation consists of several main sections as introduction, objectives, methodology, results, discussion, conclusion, and references. Moving on to the introduction. Declining of agricultural land availability is a severe issue according to ecological and environmental perspective that happens due to the land use land cover changes, urban expansions, climate change, and the coastal erosion. Therefore, evaluation of land suitability for agricultural development should be addressed for sustainable agricultural practices. Land use land cover are two separate technologies which are often used interchangeably. Land cover refers to the physical characteristics of Earth's surface, including vegetation, water, soil, and other physical features of the land, including those created by human activities. While land use refers to the ways in which the land has been used by humans. Accurate and up-to-date land use land cover information is necessary to assess the environmental consequences due to natural and anthropogenic activities. Moving on to the next slide. Soil is the foundation for all land uses. Sustainable soil use depends on three interacting factors. They are land use, soil characteristics, and other related environmental conditions. Mismanagement of land use causes degraded soil functions and characteristics. Therefore, maintaining or improving the soil quality is highly significant for the sustainability of agriculture. Soil pH, electrical conductivity, moisture content, organic matter, and the soil texture are some of the important soil chemical and physical properties which affect the agricultural production. Moving on to the objectives. The study area selected in this research is Waikal area in the western coastal region of Sri Lanka. There were two main objectives for this study. First objective was to find the agricultural land use land cover changes based on the NDVI image analysis, and the second objective was to find the spatial variations of physicochemical parameters of soil. Moving on to the methodology. Figure 1 given here represents the location of the study area, which is surrounded by the western coastal region, Mahaoya and Gingaga. This area includes approximately 8.21 square kilometers of land area. Vegetation cover of this area was determined by using vegetation indices derived from the satellite images. Then the NDVI values were calculated by using the given two equations. Accordingly, the land use land cover changes were estimated for the years 2010, 2015, and 2020. Moving on to the next slide. Figure 2 depicts the soil sample collection points of the study area. 7 locations within the study area has been analyzed to assess the soil physicochemical parameters. Overall, 42 soil samples were analyzed from these 7 locations in different depths. And these soil physicochemical parameters were identified with the following methods. Electrical conductivity and pH were determined by using the multi-parameter, moisture content by gravimetric method, organic matter content by the loss of ignition method, and finally, the soil texture was by hydrometer method. Moving on to the results section. The NDVI image analysis revealed 5 different land use land cover types, including buildings, water, open areas, vegetation type 1 and vegetation type 2. Vegetation type 1 represents shrubs and vegetation type 2 represents forest cover. According to these 3 images, it is visible that there has been major changes to the land within the past 10 years, resulting in an increase and decrease of categories. Moving to the next slide. Table 1 given here shows the numerical values for the land use land cover changes. As major changes of the area, you can see that the forest cover in the study region has decreased by 35% over the 10 years, while the vegetation type 1, meaning the shrubs, has increased by 29%. Moving on to the next slide. Figure 4 given here is a graphical representation of the mentioned changes. As you can see, the shrubs area has increased significantly, whilst the forest cover has decreased significantly. Moving on to the next slide. Table 2 gives the numerical values obtained for different soil physicochemical parameters. Moving on to the discussion. When considering the land use land cover changes, it can be stated that shrubs area or the vegetation type 1 has increased significantly from 49% to 78%, resulting in agricultural land escalation. Accordingly, as a consequence, the forest cover or the vegetation type 2 has decreased by 35% over the past 10 years, with demonstrating the consequence of agricultural land escalation. When considering the spatial variations of soil physicochemical parameters, it can be stated that the area was having neutral pH. Neutral pH favors the plant and microbial growth and also the nutrient uptake. The major soil texture group in the study area was found to be sandy loam, and it has a low moisture holding capacity. Therefore, incorporating organic matter into the soil could improve the soil structure. Soil electrical conductivity, pH, and moisture content are highly variable according to the results, and that is mainly due to the variability in the water availability. Therefore, providing frequent irrigation methods would be suitable to improve the soil quality. Moving on to the conclusion. Waikal area or the western coastal region of Sri Lanka is suitable for the agriculture with respect to the spatial variation of soil physicochemical parameters. But the land use land cover changes thoroughly implies the necessity to create new policies in the area for the protection of the forest cover and regulate the cultivations during the agricultural and economic development in order to achieve agro-ecosystem sustainability. These are the references that were used for the presentation. Thank you very much.
Thank you very much, Ms. Karunasiri, for your interesting presentation. Now the paper is open for discussion. So actually, I have one just a question for my interest. Is there any specific reason that you selected Baikal area for this study?
Hello, madam.
Yes.
In the Western Coastal region, specifically, the pollution rate is higher than any other region, and also it is very urbanized area, and also the soil erosion is very high. So to assess the effect of those things, specifically, the research was focused on this Western Coastal region.
And madam, if I add why, especially the Waikal area, I'm Sana Hirat, one of the co-authors.
Yes.
This Waikal area, when you take the spatial structure, there are very different spatial features. This area it has the Mahawaya River mouth, and for the to the Mahawaya River mouth, there are two other rivers which are flowing only inside this small patch of land, which are Lumuganga and Ginganga. So it is a very special that Gingang is not the southern part Ginganga. It is a very small river which has the same name. So this special structure of the area is very important. That's why this area was selected.
Okay, thank you. And I have another small clarification. Actually, there's a necessity of sustainable agricultural practices in the area. Can you just suggest some technological approaches that we can take for a sustainable agricultural practices? Some novel technological approaches that we can follow, just according to your knowledge.
Hello, madam.
Yes.
The conditions required for the cultivations like soil, pH, and other characteristics, they can be predicted by using the modern technologies like the GIS. And also there are various other softwares available to predict the characteristics that is required to grow the suitable cultivations in the area.
Okay, thank you. So are there any questions in the audience? In the absence of further questions, I wish to thank you, the authors, for their excellent work. So thank you. So we will move on to the next presentation. The next presentation is on an assessment of irrigation and water management services in the dry zone of Sri Lanka. This paper is authored by Sihan Saruk, Ranjana UK Piyadasa, and HMMSD Herat, and also Vajra Hettige. So this paper will be presented by Sihan Saruk. So over to you, Mr. Saruk.
Hi, on behalf of our team, I'm presenting a research paper on an assessment of irrigation and water management services in the dry zones of Sri Lanka. When it's come to the background of this study, district and divisional level officers working for irrigation and water management services are intermediaries between national level departments and farmers. They operate as facilitators and communicators, helping farmers in their capacity building, decision making, and ensuring that appropriate knowledge is applied to obtain the best result with regard to sustainable production. They usually work within communities and are expected to have wide knowledge of their sector. It is necessary for these officials to develop and upgrade their knowledge related to their day-to-day job functions. Objective of the study is, the primary focus of this assessment is to determine the current state of knowledge related to main irrigation and water management services felt by the related government stakeholders in the dry zones of Sri Lanka. Therefore, the objective of the assessment were to determine the level of subject knowledge of government officials that are required to meet their current demands for performance and to ensure that the capacity building interventions of these government officials are focused especially on the most relevant issues and the opportunities. Material and methods. Data were collected through focus group discussion using a pre-tested, well-structured interview questions during 2019. The respondents were asked to self-rank their perception of each interview questions, and choice options were coded using a Likert scale. The respondents were asked to self-rank their perception of each interview questions, and choice options were coded using 5-point scale. A modified 5-category consequence scale ranging from very low to very significant were used to categorize responses. Therefore, calculated level of knowledge perception were categorized into 5 major categories. Below 20% were considered as very low, between 20% and 40% considered as low, and between 40 and 60% was considered as moderate, and 60% and 80, between 60% and 80% was considered as significant, and above 80% was considered as very significant. Data were collected on current knowledge on following sub-questions. First one is cascade water resource development and management planning. The second one is on-farm and off-farm water management. The third one is construction supervision. Fourth one is operation and maintenance. Fifth one is finance for maintenance. And the sixth one is disaster preparedness and response planning. All these sub-questions were related to irrigation and water management. In addition to these, 27 key informant interviews were conducted with selected members of farm organizations. The qualitative and quantitative data were coded and analyzed using Microsoft Excel and SPSS. Descriptive statistics were used to analyze data obtained from a survey of purposefully selected 134 government officials from 15 government institutions and departments through official invitation from Anuradhapura, Kurunegala, Puttalam, Trincomalee and Vavuniya districts. Results and finding. The study concluded that government officials' levels of knowledge of irrigation and water management services was moderate, which is 58.9%. Our first question was related to knowledge on cascade water resource development and management planning. Within that, we had 9 sub-questions. First question was knowledge related to ancient cascade system. All the districts have recorded moderate knowledge related to that questions. And the second question was knowledge on conservation of reservations. Except Anuradhapura, all the other 4 districts has shown low knowledge related to that particular questions. And the third sub-question was on knowledge related to cascade system. Except Kurunegala, all the other 4 districts has shown moderate knowledge related to that question. Kurunegala had low knowledge related to that question. The fourth question also, the Kurunegala and Vavuniya had low knowledge. Related to fifth questions on ecosystem services of cascades, Puttalam had significant knowledge related to that particular questions, and our 6 questions was on contemporary risk to cascade and the probable impact on this cascade system. Kurunegala had recorded low knowledge, but all the other 4 districts has the moderate knowledge. And seventh sub-question was related to cascade water resource development. Almost all the district had moderate knowledge on that particular sub-questions. And our eighth sub-question was related to knowledge related to cascade management and planning. Except Trincomalee, all the other district has recorded moderate knowledge. Trincomalee had low knowledge related to that particular questions. The last sub-questions in this category was institutional role of government officials. Trincomalee and Vaunia had significant knowledge related to that particular questions, but other three districts had moderate knowledge related to that particular sub-question. Our second question was related to knowledge on water management for the minor irrigation sector. This had four sub-questions. First sub-question was on on-farm knowledge related to on-farm traditional water management system. Anuradhapura and Trincomalee district had significant knowledge on these sub-questions. The other three districts had moderate knowledge. And our second sub-question was on off-farm traditional water management system. All the five districts has recorded moderate knowledge on this. And our third sub-question was related to on-farm improved water management. For that sub-questions, except Anuradhapura, all other four districts have recorded. Our third question was knowledge on construction supervision for a village irrigation system, which had six sub-questions. Knowledge related to canal and farm road functions, earthwork, concrete technology, farm outlets, conservation of reservation, methods of turfing. Almost all the district had moderate knowledge related to these sub-questions, except farm outlets, in which Kurunegala and Puttalam had low knowledge related to that particular question. Our fourth question was on knowledge on operations and maintenance of rehabilitated tanks, in which we had 10 sub-questions. Our first question was on operation and maintenance of the village irrigation. Second question, sub-question was on flood and drought response plan. Third question was on annual working plan for operation and maintenance. Fourth question was on repair works of farm roads. Fifth question was repair works for FC canals. And sixth question was repair works of drop facilities. Seventh question was improvement for slide gates or sluice gates. Eighth question was on repair method for damaged canals. Ninth question was cleaning for water conveyance pipe. 10th question was take care of sodding grasses. So in when compared to all these sub questions, Anuradhapura had scored some significant knowledge related to some of the sub questions, but almost all the other district had moderate knowledge related to these sub questions. Our fifth question was on knowledge on financing of maintenance of village irrigation system, which had 5 sub questions. First one was related to financing of village tank maintenance, in which Kurunegala district had shown significant knowledge, and Anuradhapura and Vaunia had moderate knowledge, and the Puttalam and Trincomalee had low knowledge related to that particular sub questions. Our second sub question was on developing and maintenance of developing and implementing financing plan for village irrigation system. For that particular sub-questions, Vaunia had significant knowledge. All the other 4 districts has recorded moderate knowledge. Our third question was related to knowledge on management of funds. For that one, Anuradhapura and Kurnagal district has recorded significant knowledge. And our fourth question was related to accounting and bookkeeping. For that one, Anuradhapura, Kurnagal and Puttalam district had moderate knowledge, and Trincomalee and Vaunia district had significant knowledge. Our fifth question was ensuring transparency of funds management. Almost all the all 5 districts has recorded moderate knowledge related to that particular sub-question. Our sixth question was related to knowledge on disaster preparedness and flood response planning. For this one, we also had 4 sub-questions. First sub-question was on village disaster preparedness, and second question was related to flood response for village irrigation system. Third question was related to drought response for village irrigation system. And fourth question was operationalization of preparedness and contingency plan. For these sub-questions, almost all the districts have answered, almost all the districts had moderate knowledge, except drought response for village irrigation system. For that one, Anuradhapura district has recorded low knowledge related to that particular sub-question. Conclusions. More emphasis should be given to introducing practical tools and methodologies for cascade management planning to expand the capacity of government officials to deliver their roles on cascade water resource development and management planning in the dry zones of Sri Lanka.
Thank you very much, Mr. Saruk, for your interesting presentation. Now the paper is open for discussion. So I wish to ask a certain explanation. Like this is a general question. Can you just explain the adverse impacts on national economy with this low-level knowledge on environment or knowledge on irrigation and water management of the relevant officials?
Sorry, can you repeat the question, please, Madam?
Yes, I just want you just to explain the adverse impacts on national economy with the moderate level knowledge of the irrigation and water management officials.
Yes, actually, this is impacting in several ways in terms of, economic terms, especially most of the private sector companies involved in this process has gone far away. If our officials, especially the extension officials attached to the agriculture sector and irrigation sectors are not aware of these modern technologies, that will be, definitely creates a lot of impacts to the economy. Say, for example, even though the water resources is free of charge, sometimes it's a cost for our economy. So there are a lot of technologies introduced to save water management in this area, especially in the dry zones. If these government officials are not aware of those technologies, unnecessarily will be, wasting our resources.
Okay, according to what you have found, this is just for interest, I'm asking, what was the cause of this low level knowledge? Where was the issue?
One of the main issue we found during our assessment was especially these new knowledges to be passed to the government officials who are directly interacting with the farmers. But unfortunately, some of the officials who are directly working with the, say, I'm referring to the Kupanisa, they are attached to, not attached to the Department of Agriculture, because the knowledge flow is directly coming through that line, but they are mainly attached to the Department of Agrarian Services. So that linkages between the Agriculture Department, the Department of Agrarian Services need to be established. The other thing is some of the forecast that is generated from the agriculture departments are, not directly reaching to the farmers since there is a, mismatching of these channels.
Okay, so have you thought of forwarding your findings to the relevant authorities for their attention?
Yes, actually, we have shared this finding with the relevant authorities and the our funding agencies. So they will be most probably they actually use these findings to start the training program for these government officials.
Okay, thank you very much. Are there any questions from the audience? So in the absence of any questions, I wish to thank Mr. Saruk and the team for their interesting work that they have done. So thank you. So now we'll move on to the last presentation. The presentation will be on the impact of COVID-19 pandemic on surface water contamination due to disposal of plastic protective equipment and discharge of wastewater. So the paper is authored by T.A.N.T. Pereira, A.I. Madhumali, S.H.L. Sandamali, P.P.A. Sanjeevani, S.M.F. Ramashana, and Kanjana U.K. Piyadasa. So the paper is presented by T.A.N.T. Pereira. Ms. Pereira, over to you.
I am Nilusha Pereira from the Department of Environmental Technology, Faculty of Technology, University of Colombo, here to present the impact of COVID-19 pandemic on surface water contamination due to the disposal of plastic protective equipments or PPE and discharge of wastewater. This presentation contains with introduction, objectives, methodology, results and discussion, and conclusion, and then the references. Due to the rapid spread of coronavirus since 2019, most of the countries took the decision to lock down the countries and people started to self-quarantine and stay home. So roads have a less number of cars, vehicles, and also industries are closed. Therefore, it seems that there have been environmental benefits with decreased environmental pollution due to this decrement or the reduction of industrial activities and the anthropogenic activities, but there were other consequences due to improper disposal of plastic protective equipments and also wastewater. Almost all plastic protection equipments like mask, face shield, hand gloves and goggles are made by using different kind of plastic. So these plastics most often being disposed in everywhere, and there might be number of PPE around the drainage systems and canals and also reservoirs. So this improper disposal of PPE and wastewater caused escalated water pollution during COVID-19 pandemic period. Therefore, this study was focused on assessing the water quality changes and assess the human perception on water pollution during COVID-19 pandemic. The main objective of this study was to explore the immediate impact of COVID-19 safety processes on surface water bodies with these two specific objectives. to study inland water quality changes during COVID-19 pandemic Sri Lanka, and also to determine the effects of municipal wastewater discharge on surface water bodies during COVID-19 pandemic. So the present study is a national level cross-sectional online survey conducted using Google Form web survey platform from 27th May to 2nd June 2021. The country lockdown for the third COVID-19 wave in Sri Lanka commenced on 21st May and lasted during the period of data collection. And the online questionnaire was specifically built using Google Form. The questionnaire was available in three main official languages in Sri Lanka: English, Sinhala, and Tamil, so that the participant could select the preferred language. For this study, demographic data, water body changes, and waste disposal related data were used, and reliability of the adopted questionnaire was tested through piloting prior to survey administration. All the variables were analyzed qualitatively and were expressed as a percentage and numbers. The descriptive statistics were employed to explore the demographic parameters of the study sample, and results were presented as frequency and percentages in percentage for categorical variables, or mean and standard deviation for continuous variables. According to the results of the study, participants represented every district in the country, and the majority of respondents were in urban and suburban areas. It's about 66.6%, and the survey was accomplished by 466 contributors, and 81% were in the 18 to 35 age category. Among these participants, 63% were female and 36% were male, and one respondent did not actually disclose their gender. According to the findings of the study, 44% of respondents have been noticed increment in water quality and the water pollution, and 27% were noticed reduction of water pollution during COVID-19 pandemic, according to this figure, and 29% of respondents have not been noticed any changes in water pollution during COVID-19 pandemic. Among the respondents, 50%, approximately 50% were mentioned that the water pollution in their region were increased due to the human activities, and 28% mentioned it was because industrial activities. However, during the COVID-19 pandemic, decrease of water pollution has not been significant. As I mentioned, most of the respondents mentioned that disposal of PPE, plastic protective equipment, was the reason for the increase of water pollution in their region, and also at least one face mask used by the people or the respondent per day, and it was 43% and 36% of respondents were used 2 face masks per day. That means the usage of face mask or the PPE was increased during this COVID-19 period. According to the online survey, most polluted water sources were rivers. It is about 27%, and then reservoirs 26%, then culverts and the streams. So, and it has been due to higher use of PPE during the COVID-19 pandemic. According to the study, 49% respondents have been used PPE than previous. Actually, during COVID-19 pandemic, the PPE, these face masks, face shields, gloves, and etc. So usage have increased ever than before, and therefore the disposal of plastic into the environment have increased. So every respondent use at least one face mask, as I mentioned, per day, and more than 50% using greater than 2 face masks, according to our study. When considering wastewater discharge during this COVID-19 pandemic, actually it was also changed with this pandemic period, because most of the people are using used hand sanitizer and different kind of cleaning agents. Therefore, we analyzed the type of cleaning agents that were used for hand washing and the kind of things during this COVID-19 pandemic. So more than 40% respondents were used normal soap, bathing soap, for their cleaning purposes during this period, and also hand sanitizers with less than 60% alcohol were used by 16% of people, 16% of respondents, and hand sanitizers with more than 60% alcohol were used by 22% of people. Therefore, different kind of chemicals and alcohols are included in the wastewater they discharge from their home or their institution or any places they live. So the wastewater discharge methods during this COVID-19 period was also have some changes. So most of the people, around 55% respondent were discharged wastewater separately, but around 90% of the people discharge wastewater as usual. So this could be a major problem for the water pollution in during this COVID-19 period. Most of the respondents were directly discharged wastewater to the canals. It's about 52%, and then 17% of the people discharge. They didn't have any idea regarding this wastewater disposal method. As conclusion, overall results of the study reveal that plastic protective equipments contribute to the growth of water pollution during the COVID-19 pandemic, and the amount of plastic protective equipments in water bodies have been increased more than usual, and the most polluted water sources were river. Before the pandemic period, the use of face masks was limited to specific individual like health sector, but in this situation, the general public also had to use face masks daily for their protection. So preventing PPE release to the environment that have the potential to discharging microplastics and ecological stresses into the aquatic water bodies to evade the deterioration of the water quality. Overall, significant water pollution due to increased usage of PPE was observed, and proper disposal methods should be implemented while making aware public community. These are some references we use for this study. And thank you very much.
Thank you, Ms. Pereira, for that interesting presentation. Now the paper is open for discussion. So, okay, now in the presentation you mentioned that you used a questionnaire to find out about the whether there's a water pollution caused by this use of the personal protective equipment. What sort of pollution that the respondents spoke about?
Actually, madam, we mainly focused on the plastic waste which used during this COVID-19 pandemic, like face mask and face shields and the things. So most respondents were respond about the face mask usage in their regional areas. So most of the respondents were responding that face masks are mostly discharged into the culverts and the reservoirs in their neighbors locations. So that is the most significant one we collect from those questionnaire survey. Actually, this questionnaire survey were conducted in the in island wide actually. So most of the region in southern province, most of the urban areas, they mention about the significant changes of the reservoirs and the rivers and also in lakes were due to this face mask accumulation.
Yes, I just, can you just tell me what sort of changes in reservoirs and other water bodies that they have observed?
Actually, due to the accumulation of this plastic protective equipments like face masks, respondents will notice that some what we call turbidity changes, like the changes of the color of rivers, like that. So but it's not significantly we recognize through this questionnaire survey. We have to go for the next phase of the study to identify using image analysis, satellite image analysis. So most of the animals like birds and fish, they were contaminated with this face mask.
Okay, according to my views, that if you use the word the inland water quality changes, you have to connect that with the physicochemical characterization of water, and especially with the discharge of these used personal protective equipment, that is basically the face mask and all that. There might be some kind of bacterial contaminations that are coming with those. So you have to conduct the microbiological analysis in order to, I think, use the word the water quality changes. And also, I just want to mention one thing, that when you use PPE normally stands for personal protection equipments. And if you are focusing on plastic based personal protective equipments, then you can say it's plastic personal protective equipment, I think.
Yes, madam. In this survey, we mainly focused on only the plastic protective equipments, because in personal protective equipments, it's contain like some other garments and earbuds as well. So here we mainly focused on face masks, face shields, and goggles only. That's
okay.
why.
Okay, then it's better to mention like plastic personal protective equipment.
Yes, madam.
Okay, thank you very much, Ms. Pereira, for your presentation. So are there any questions from the audience? So in the absence of any questions, I wish to thank Ms. Pereira and team for their interesting work. And that marks the end of this technical session. And in this technical session, there were 6 presentations. And these 6 presentations focused on different areas of environmental technology. And the research outputs were very interesting. And I thank all the authors who has done a very commendable work actually in this research areas. And also I invite you to submit abstracts for our next symposium as well in year 2022. So thank you very much.
Thank you very much, Professor Shamini and Dr. Priya Tilaka, for chairing the session. Now we are about to start the technical session 3. I would like to call Dr. Rohana Vijayawardena and Ms. Chatrika Silva from the Department of Instrumentation and Automation Technology to chair the session.
Thank you, Dr. Ansika. Good afternoon, everyone. So I warmly welcome you all to the technical session 3. We have 3 presentations for this session, and I will be chairing first presentation, and other presentations will be chaired by Ms. Chatrika Silva. Each presenter has 10 minutes to present, and at the end of the presentation, there will be 5 minutes of question and answers. Our first presentation is Smart COVID Assist, continuous monitoring system for the COVID-19 quarantine patient using mobile app in IoT environment. NR Samarasinghe, AHM GB Abesinghe, and NM Tilakaratna are the authors of this presentation. I would like to invite Dr. Rohan Samarsinghe to deliver the presentation. Over to you, Dr. Rohan Samarsinghe.
COVID Assist, a remote monitoring system for COVID-19 quarantine patient and contacts using the mobile app in IoT environment. I am Rohan Samarsinghe from Department of ICT, Faculty of Technology. COVID-19, which is a deadly virus, was first diagnosed in 2019 in Wuhan, China. Since then, it has made a severe damage to the world, having more than 253 million patients and 5.1 million death toll. Corona is seriously threatening to the human health and as well as the economy of the countries and the world. When we consider the Sri Lanka, mainly SARS-CoV-2 lineage B1.1.7 originally detected in UK variation and B1.617 Delta and Delta Plus variation first detected in India could be identified from the Sri Lankan patients. So when we consider this prevention method, different technologies are being used with the experiments to diagnose and identify and prevent from spreading the COVID-19. Some of the technologies are deep learning, big data and data science, GIS, ICT and IoT technologies are being used. These technologies can be used to diagnose high-risk COVID patients using the CT images and clinical data and visualize of epidemic and epidemic information, spatial tracking of confirmed cases, areas, etc., and design the lockdown cities and areas. There may have some